So let's get started. Everybody take a seat. So my name is Tobias Weiss. I'm a physician scientist here from Zurich. And it's a great privilege actually to welcome you all on behalf of the program committee, comprising Markus Mann sitting over there, Chiara Magnani sitting over there, Burkhard Becher sitting over there, including myself, to this 10th Symposium Zurich Immuno-Oncology. It's nice, it's the number 10, and actually if we look back 10 years of immuno-oncology, in 2016, that was the year when the American Society for Clinical Oncology named immunotherapy as the advance of the year. It was actually the first checkpoint inhibitors have been improved. But since then, there are really numerous innovations, breakthroughs, and we gained a lot of scientific insights. And it's really exciting to hear from a fantastic lineup of international outstanding speakers about these developments. So a few remarks. We have over 600 attendees, many actually hybrid, 260s are supposed to be in the room. It's at the edge, so it fits 300 people, so that's perfect. And we really also have sponsors shown over here, and we would like to thank the sponsors, of course, for support. We would also like to thank Silke Uflecker sitting over there, Christopher Mertens sitting over there for technical support. So with all of you, this would not be possible. And then we remind the speakers to keep in time, to have also a dedicated time for questions. Also to the chairs, please pay attention, particularly for the flash talk people. So they have the challenging task to present a topic in 2.5 minutes. So please stick to the time. With this, I really would not like to lose more time and hand over to the first chairs. One more thing, you see these metal boxes in front of you, these silver things. So if you basically open these are microphones. So if you have a question, open the box, press the button, you will see a light, and then speak to the microphone. Go close there, switch it off, and close the silver box, right? If it's not working, don't worry, we have hand microphones. So with this, I hand over to Andreas, Wicki, and Anne Müller for the first session.
Yeah. No. Okay, well, that's different. Okay, so my name is Andreas Wicki. I'm the clinical director of the Tumor Center, so a warm welcome from my side. Also, I'm very happy we have so many attendants this year in this symposium. That's fabulous, and we have a range of really outstanding speakers, so I'm sure this is going to be a lot of fun today. So basically immunotherapy, for many decades people tried to tackle it and they were unsuccessful, and the checkpoint inhibitors were really the first immunotherapy drugs which made it to the clinic and provided overall survival benefit to our patients. And that's why we have dedicated the first session of this symposium also to checkpoint inhibitors, and then afterwards we're going to move on to many other topics in the field of immunotherapy. So it's my pleasure and privilege to introduce the first speaker of today's symposium, that's Sérgio Quezada from the University College in London. His group focused on mechanisms of checkpoint inhibitors, and there's also a strong focus on regulatory T cells. I'm very curious to hear about your thoughts.
Thank you very much. And I want to thank the organizers for the invitation to this stellar meeting. Really, we're talking with Burkhard, and the lineup, excluding myself, it's quite impressive. So I'm really looking forward to a great day. Yes, so my lab has been working on checkpoint inhibition and mechanism of action of antibodies targeting immune checkpoints for a long time, and also regulatory T cells. And I am not going to talk about any of that. I'm actually going to talk about work from a very talented postdoctoral fellow in my lab, Dimitris Garifallos, who came to the lab wanting to screen for novel potential relevant immune checkpoints for immunotherapy of cancer. And he wanted to set up a classical genome-wide library screening, trying to look for depletion or enrichment of different guide RNAs. Again, on a genome-wide type of approach. And he wanted to do this in vivo. So he set up several experiments in the lab using a mouse model of colorectal cancer. So these are transplantable tumor cell line, MC38, very classical, nothing special about it. But he generated this huge library of guide RNAs targeting the whole genome and tested for enrichment or depletion of different guides, both in vitro. Can you guys see the mouse? Yeah, in vitro and also in vivo, which was the important part. So when he ran that library, he found several guides that were enriched and several that were depleted. Amongst the ones depleted, there were these two molecules, MET25 and MET16, that kept on coming on all the repeats that he generated. So one of the key things he wanted to do, though, it was also to evaluate what happens when he takes the shortlisted guides from the first system, and he tests the validity, still the depletion or enrichment of those guides in a system that was immunocompetent versus a system that was immunodepleted. So what he wanted to do was not only pathways or identify pathways that affected tumor growth, but that affected tumor growth in the context of an immune system. So that was the goal. And for that, he used, again, C57, implanted the cells into C57 Black 6 or into immunodeficient animals. And the guy that kept on coming, or the target that kept on coming and actually quite exacerbated, was MET16. So what is the MET16 protein? So MET16, it's a small component, it's a small subunit of this large mammalian mediator complex. The mediator complex, it's linked to control of RNA transcription. So it interacts with RNA polymerase II. It's a co-activator complex. And what was known to us, completely ignorant in this field, was that it gets activated in response to stimuli. And it's a global controller of transcription. So, well, quite a complex molecule, but does it play really a role on immunity? That was his question. So the first classical experiments you do when you have an interesting guide that is coming up over and over is to, make a cell line that is just knockout for this particular target. So he's generated several tumor lines, again, all mouse, that were either knockout for MET16 or they have control guides on it. And one of them was the colorectal cancer cell line, where he generated the initial screen, the MC38. And what you guys can see here is that if you implant into immunocompetent animals, MET16 knockout MC38 tumor cells, the mice survive longer than if you do it with control guides or if you do the same experiment in an immunodeficient animal. So there's something about knocking out MET16 on tumor cells that it's activating sort of an immune response that is preventing that cell line to grow. If you do this with lung cancer cell lines planted in the lung, the result is actually more striking. MET16 knockout lung cancer cells cannot implant at all. They don't grow at all in this immunocompetent mice. And again, if you do it in immunodeficient animals, you don't see any sort of effect. So what is MET16 doing in vivo? So we did the classical mouse type of experiments. You take an animal, you challenge with the MC38 cell line in this case, or the KPV6 with the different ones. And then after different time points, you run flow cytometry or transcription analysis. And when I'm just going to show you for a matter of time, just showing you the single cell RNA-seq data. And basically, when you investigate the tumor microenvironment of these animals, you find all your classical favorite subsets, right? Effector memory, naive T cells, TPEX, exhausted cells, etc. But what happened constantly is that this cluster of effector memory cells is the one that we saw significantly increase every time you had a MET16 knockout tumor cell line. So MET16, implantation of MET16 knockout cells into a mouse generates a large expansion of the effector memory population. So this is not reversing exhaustion or anything like that. It seems like it's particularly the effector memory CD8 compartment, the one impacted. We didn't see a lot of differences on the CD4 compartment. What we always do in the lab is, if we're doing single cell transcriptional analysis, we link it as well with TCR. That allows us to look at clonal relationship and try to understand a little bit more in an antigen agnostic manner, without having to put over albumin in our tumor cells, whether we think or not we have a clonal response. And indeed we did. We observe clearly that in the effector memory population, there is a significant increase of specific T cell clones, suggesting that there is further antigen engagement and probably priming. When we do relationship analysis in terms of clonal relationship analysis, the vast majority of these effector memory cells that are expanding, those TCRs were found in naive T cells in the untreated animals, suggesting that there is a lot of de novo priming. And again, that's quite interesting. I think I should also mention, relevant for this, that the NC38 model is a very immunogenic tumor cell line. Meanwhile, the KPV6 lung cancer cell line, it's a line that's not immunogenic at all. It's really hard to get protection against that tumor type. So everything was pointing to CD8 activity. So we did the classical experiment that you will do. You challenge your mice with your control cell line or your MET16 knockout tumor cells. And then at different time points, you do a depletion experiment. So we tried depletion of different subsets. I think that there is a combination of factors. Depletion of NK cells did have a partial impact on tumor control, but it was consistently the depletion of the CD8s that make you lose protection. So CD8-mediated immune response induced by lack of MET16 on the tumor cells. So what happens beyond the T cells, right? So we're observing a change on the T cell compartment, but in reality, the cell that we're knocking out or knocking out of MET16 is the tumor. So what happens to a tumor cell? So this is an ex vivo analysis of the tumor compartment at the single cell level as well. And what we observe consistently is that when you knock out MET16 and you look at the cells ex vivo now, the tumor cells, there is a huge increase in type 1 and type 2 interferon signatures. So it seems that by knocking out MET16, your tumor becomes intracellularly very hot and very sensitive to interference, both type 1 and type 2. And the subsets that are the most sensitive to interference are the cells that disappear the quickest upon immune engagement. And in fact, so this is just some cell chat analysis, just trying to look at your knockout versus your control tumor cell lines and what are the clusters that change. And C0 and C1 are in the previous slide, are the populations that disappear the most. And those are the ones that seems to be in strongest communication with the effector memory compartment. And in fact, among the strongest interacting pathways, it's MHC and antigen presentation, the one that appears, the one exacerbated. So you knock out MET16 on the tumor cells, they sense more interference, and it seems like they are able to present more antigen, at least by this transcriptional analysis, to the CD8 compartment. Based on those changes, we did a lot of analysis, and one of the things we look at is early transcriptome analysis using TT-seq. And out of all the transcription factors that get upregulated, the ones that are significantly upregulated once you knock out MET16 is interferon response elements. Again, an significant intracellular change on these cells. We thought that eliminating MET16 because because the mediator complex is a global controller of transcription was going to change things in a unspecific manner, but it seems like everything that we're observing changing has a relationship with type 1 and type 2 interferon. And in fact, we started doing experiments in vitro now where we try to understand transcriptionally how did this change happen and what type of genes were observed. So we started running controls against interferon gamma stimulated tumor cells. And what we observe is that the vast majority of the genes upregulated when you knock out MET16 in a tumor cell are the same or similar, at least overlapping, with genes upregulated when you treat cells with interferon gamma. And as you guys know, interferon gamma is an essential cytokine, quite effective, used by the immune system at attacking tumors. So when we neutralize either interferon gamma or type 1 interference, we completely lose the in vivo effect of MET16 knockout, by closing that loop and saying, yes, there is higher sensitivity or something happening at the level of interference. And when you eliminate those, both in a cell intrinsic and intrinsic manner, you lose tumor control. So we believe that there is this increased sensitivity to interference, the one leading to a higher potential antigen presentation, higher T-cell response, and then tumor elimination. So that's quite interesting. Now, what could be triggering this increase in tumor immunogenicity? And that's when we started going a little bit deeper and beyond our comfort area of expertise. So one of the things we decided to look, because of this large intracellular change on type 1 interference sensitivity, we decided to look at whether there were changes on the expression of endogenous retroviruses. So one of the things that the mediator complex could be doing, it's controlling endogenous retroviral expression. And in fact, we observed that either the MET16 knockout tumor cells or the MET16 knockout tumor cells incubated with very little interferon gamma have a significant de-repression of endogenous retroviruses. And that now started making sense. Although we're doing the experiment, we haven't completed this. This is an ongoing unpublished story. Well, although we haven't completed all the mechanisms, what we think is that this intracellular de-repression of ERVs, it's leading now to increase in CGAS and intracellular sensitivity of RNA. And that's leading to this super activation or increase in immunogenicity of the tumor cell. So we think that this is one of the key mechanisms by which you are converting these tumors into hotter, more inflamed or immunogenic tumors. Now, what's quite interesting is that we observe this change, right, this increase in line elements, which are known to produce a higher level of immunogenicity intracellular. But then when we start looking a little bit further, doing sequencing, again, RNA sequencing, but also exon sequencing of the tumor cells, we also observe a significant de-repression and appearance of alternative splicing elements, right? So in addition to have an innate change, like innate immunity change on your tumor cells, now actually your tumor cells are starting to express a lot of neoantigens, right, based on alternative splice element. And in fact, we just run the in silico experiment of taking all the skip exons that are appearing once you knock out MET16 in whatever tumor cell we use, and we model potential binding to the class 1 alleles of the mouse. And basically, when you either knock out MET16 or knock out MET16 in presence of interferon gamma, we see more than 100 neoantigens predicted to bind with high affinity to the mouse HLA, the mouse MHC. So MET16 quite impressively not only regulates innate immunity in a tumor cell, but also the ability of that tumor cell to present antigens. And that's what we believe that this is a checkpoint, or we are calling it in the lab as a checkpoint of immunogenicity. Now, so far, we are at the point of showing interesting data that you knock out MET16 and you get this amazing immune response, even in very cold tumors like the KPV6 lung cancer model. But what do we know about humans? So in human cells, we've been doing the parallel experiments, and what I can tell you is that MET16 has the same impact in terms of innate immunity, so activation or higher sensitivity to type 1 and type 2 interference, and also the increase on alternative splice elements, so also increasing immunogenicity. So we started running experiments now using human tumor cells and human T cells targeting a model antigen, in this case, and NY-ESO. And when we incubate, when we do co-culture assays, and we looked at activation of the T cells, we see that MET16 knockout human tumor cell lines now induce a much higher activation of effector T cells by looking at CD137 expression or CD125 or IL-2 receptor alpha upregulation. And when we do, again, on those co-culture assays and we track tumor cell death or tumor cell killing, we also observe that the MET16 knockout human cell lines are much more sensitive to NY-ESO TCR transgenic T cells. So where we're going with this story now, it's the more challenging bit. Is this is an intracellular protein, right? And targeting intracellular proteins is not an easy task. We initiated a degrader campaign with a company that's well known called Wicki, and we already designed a set of MET16 binders. And the idea is to link this to E3 ligases and induce and create this PROTAC type of proteins. How are we going to be delivering them into tumor cells? We believe we're going to take an approach using LNPs, but that's all work in progress. So in summary, we have established an in vivo system for identification of potential targets for immunotherapy. And we got lucky enough to find this MET16, this complex, which we believe is quite a promising target for immunotherapy. And where I see the value of this is in actually converting cold tumor cells into hot tumor cells, right? So through this type 1 interference sensitivity, but also to the expression of neoantigens. There is some emerging data in the lab that actually, if you fiddle with MET16 expression on T cells and on macrophages, there is also a change, and a change that is beneficial, but that's work in progress. All the work that I show you is mice related, but the human data, it's panning out in the same direction in terms of knocking out MET16, increases immunogenicity of the human tumor cell lines that we have generated, both colorectal and human lung cancer, and it gets more antigen presentation and better killing by T cells. So with that, I wanna thank you for your attention. Again, the organizer for the invitation and the amazing team that I have. This is us, our last lab retreat. Dimitris Garifalos had led this from the beginning and put together a team of collaborators within the lab that had really been complementing his expertise. He's a guy that is an expert on screens, not an expert in cancer immunology per se. And I think it has been the combination of him being in the lab and in collaboration with lots of people that are on the bioinformatics side, on the wet lab, on the mouse side, that has led to this phenomenal collaboration and work. So thank you very much for your attention.
Okay, so thanks very much for this very interesting mechanistic presentation. Interestingly enough, for PRO-TACs, we've seen the first trial to degrade estrogen receptors published last year. So the concept itself seems to hold true. So it's very interesting. Questions, Marcus?
Yes, wonderful presentation. I have two questions. The first question is, if you take these effector memory T cells from the ones that arose in the MET16 knockout tumors and transfer them to a situation where they meet MET16 competent tumor, are they able to eliminate?
That's a great question. No, so we haven't done that. That's a phenomenal question. So we're definitely going to do that when we go back. Now, what I asked the team to do, though, perhaps going in the same direction, is that I want to generate more human data, right? I think the mouse data is great and it's mechanistic, but I want to generate more human data. So what we're doing is we have a large panel of tumor cell lines and organoids from colorectal cancer, lung cancer, human colorectal, lung, and brain cancer. So I want to put MET16, knock out MET16 in all those lines and look, one, at potential overlap on antigens being arising. So is there an antigen that is being shared that we are unveiling, or multiple antigens, or are these completely personalized antigens for that tumor cell? So I think that's a question we're trying to answer with the humans, but the mouse experiment, it's a good one. Thanks.
And the second brief question is, if you have an inducible MET16 knockout, would they get autoimmunity?
Yes. Yes. So this is going to be, so our biggest challenge will be to find the therapeutic window for this drug. So we are completely acknowledging that. And I think that this is a therapy that needs to be a local therapy. And that's what we're thinking about LNPs.
More questions? Yes, I guess you were first.
Thank you. Thank you very much. Very interesting.
Thanks, Erin.
I had the exact same question as you about transferability with the T cells. I think it's very important to do. The other question is, have you looked at in human if you have some deletion in the tumors of these molecules?
So no, we haven't. So yes, we have looked into humans. We haven't found any specific deletions or amplifications of the gene. So that's one of the things we're looking. Are there amplifications in tumors that are more resistant to immunotherapy? We have looked at our CPI 1000 cohort and now the 2500 that we're working on with Charlie, and basically there is a correlation on the expression of MET16 and how hot or cold the tumor is. We're looking at it a little bit further. In terms of the transferability of the immunogenicity of the T cells, What I find fascinating is that even if if it's not transferable, right? So we're all talking always about personalized therapies in terms of targeting neoantigens, etc. Here you have a drug that can increase that personalization, but it's the same drug, so you could give it to any patient, right? So it is quite. That's why we find this target quite interesting, and we're going quite aggressively after it.
3 more questions. The first, then you have Burkhard and then Anne. Yes, please.
Yes, they do fantastic work. I have a question pertaining and relates to gliomas. So whenever we see also with epigenetic treatment ramp up of CAR-T pathway, PRS, so one of the mechanisms that always pops up is increase of chemokine CCL9, CCL10 attractive T cells. Does this play a role in your system? Do you think that this is part of the mechanism?
I think it is, but we don't have the data. So on that panel of tumor cells that are being knocked out, we're looking at everything that is being secreted by the tumor, and obviously on the single cell RNA-seq, we're digging a little bit deeper to see what it does. One of the things that I'm asking Dimitris to do is to knock it out on our mouse models of glioblastoma and get more information. That would be amazing.
Yeah, super exciting, and I love the screen idea. This is very, very clever. And MET16 looks like a great molecule to be working on, but there are 2 things I didn't understand. Number one, the effect that you have in the NSG is spectacular, no question about it. But the T cell depletion is rather mild effect. So I'm wondering why you pin the mechanism exclusively towards killing interferon gamma. And the other question briefly asked right away would be, so the good Lord probably didn't invent MET16 only as a tumor antigen. So what is its natural function, maybe in immunity as well?
So the natural function of immunity of MET16, that's something we want to find out. We haven't found much in it. Dimitris always puts a bullet that I always delete on his presentations that it has been implicated in a plant immunity. Maybe, right? But yeah, that's something we need to find out. And then in terms of the CD8, so if you deplete CD8s, you completely lose tumor control. There is delay, but there is still 100% of the mice die. However, there is a role for NK cells. So NK cells are also participating there. So it's not only CD8s.
Anne?
Very nice presentation. Given that this mediator complex is so big and only 1 or 2 of the components were actually I think there are tumor-specific versions of the mediator complex that are not shared by the normal cells.
Yeah, so I didn't have time to show all the work, and there is a lot of work done in collaboration looking at chip analysis and seeing what else is changing, what is tumor-specific versus non-tumor-specific. In our screen, it was MET16 and MET25. We find out that there is another group working on this, and they came out with there is another MET that is also funny. So it just happened that in our screen, those came out as the highest targets. What I didn't talk about is that there is data from yeast that actually suggests that by taking down MET16, we might not be taking down the whole complex, but actually there is a subpart of the complex that remains with activity. And that's something that we need to find out more.
Question from Chiara.
Yes, there were some studies recently published with the screen more or less similar to what you have run on CAR-T cells, showing that the mediator complex, not specifically the MET16, but the mediator complex is very important in the function of CAR-T cells, and if you knock out, they are working very well, and they upregulate multiple transcription factors. Do you think there could be a synergic action in blocking the mediator, both at the tumor and the T cell level?
Yeah, so unfortunately, that study scooped one of my collaborators. So when we started working on this, a collaborator of ours said, oh, MET16, we have something very similar but on T cells. So actually, if they're knocking out the same MET16 molecule, they have better activation of T cells and better killing in vitro. And actually, the CAR-T cells resist quite well to serial target engagement. And that's what we said, well, we're definitely going after, and we want to go after with a drug, because we think that everything that we've done, right, is intrinsic to the tumor. If we go with a drug, we'll have actually the double whammy of targeting the tumor and the T cells. And as I said, we believe that because of these changes, we might also be affecting the myeloid compartment. But that's why we need a drug to start investigating those things.
Okay, we'll take one last question because we are running out of time, please.
This very nice presentation. I had a question regarding this massive wave of alternative splicing that you see when you lose the MET16. So are you only generating new antigens there, or could it be that this splicing just strikes other essential genes, maybe for antigen presentation or any other genes that you have not?
That's a great question. So there is transcriptionally, the tumor cells are like some of the pathways are the most upregulated related to antigen presentation. I don't know whether that's a direct consequence of some new splice variants. It could be, but definitely I think the splice variants what are generating are new antigens, and that's one of the. So we're doing immunopeptidomics now to try to figure out what's coming out on the HLA.
Okay, so thanks again. Very interesting presentation.
Thank you very much.
So I want to apologize to the people whose question I couldn't take. I realized there was also online questions, but the time simply doesn't allow for that. So it's a pleasure to introduce the next speaker, Professor Lukas Flatz. He's the head of dermato-oncology in Tübingen, and your group is also concerned into looking what the connection between autoimmunity and, on the other hand, the effect on the tumor is. So I'm very interested to hear you talk.
First of all, I would like to thank the organizers for having invited me to this wonderful meeting. And we already have heard it today that in skin cancers, particularly melanoma, checkpoint inhibitors change how patients survive. And we have already the data for 10 years. And we know that 50% of patients are surviving, albeit they were diagnosed with a stage 4 melanoma. But this comes at the cost. And what we can see on that slide is that basically almost half of the patients developed immune-related toxicity. And on the right-hand side, you can see that up to 1.2% of patients develop fatal toxicity. And me as a dermatologist, I observed, and you can see that here rash and vitiligo is a quite frequent side effect of the treatment. Often it's a bit overlooked because it's usually not life-threatening. But what we observed is early on that even if the patient has the same treatment, for example, the anti-PD-1 pembrolizumab, in one case, the patient receives it for a non-small cell lung cancer, the skin toxicity looks differently than the skin toxicity from a melanoma patient. Or in other words, vitiligo, meaning that you get an attack of the healthy melanocytes in the skin. You rarely see in a lung cancer patient, but you frequently see in a melanoma patients. Moreover, vitiligo is the immune-related adverse event which most strongly correlates with improved overall survival, as you can see on that graph here on the right-hand side. On the other side, if you go into the literature and look for vitiligo patients, so if you have a patient with vitiligo coming to us, when I see a patient for the first time with vitiligo, I need to rule out that this patient has somewhere an unnoticed melanoma. However, if this patient then was not diagnosed with a melanoma, the risk to develop a melanoma is much lower than in the population without vitiligo. So that already tells us, and you see it in the title, that there must be something like protective self-reactivity. And the goals of my talk are now to decode autoantigens leading to checkpoint inhibitor-mediated autoimmune diseases, and finally, to exploit tumor-associated autoimmune responses as cancer treatment strategies. And I would like to emphasize that we know that for several treatments, we already target kind of lineage antigens. If we have a CAR-T cell targeting CD19, that's a lineage antigen. If we target HER2 using trastuzumab, either naked or combined with deruxtecan, we are also targeting kind of a lineage antigen. But in solid cancers, many good or many diseases don't have a good antigen, and maybe this can help us for that. Let's start again from the melanoma side. So we have then shared antigens between melanoma cells and melanocytes because these antigens or proteins are relevant for the melanin production. And two good examples are GP100 or MART1. Of note, these antigens are also used from the pathologist to determine if it's a melanoma. And this has also been used for T-cell engagers. So this was basically the first-in-class T-cell engager, so a soluble T-cell receptor targeting the GP100 antigen. So again, melanocyte differentiation antigen, and this, as a second arm, it has an anti-CD3, and it got approval for the orphan disease uveal melanoma, and right now the clinical trials are running for cutaneous melanoma. However, you see it at the bottom, tebentafusp and on the T-cell engagers that are based on soluble T-cell receptors, they are limited by the HLA restriction. In tebentafusp, it's HLA-0201. So in our audience here, maybe half of the people are HLA-0201 positive. And as I said, we observed that lung cancer patients, and if I talk about lung cancer, I mean non-small cell lung cancer, we realized that they have kind of a lichenoid rash. You see an example here in the middle. And you can also see that these patients, they have a good response to checkpoint blockade. And we then thought we should set up a melanoma cohort and a non-small cell lung cancer cohort with patients showing skin toxicity. And starting from this skin toxicity, we would like to decipher antigens which are involved into that. And interestingly, these patients with skin toxic effects have also a better overall survival. And what we could then observe is when we plotted the adverse events, you can get that from the FAERS database, with the tumor to tissue similarity score, we could see that the more closely the organ is to the tumor, the more likely it is that toxicity will attack that organ, which of course makes sense. In lung cancer, you could frequently get pneumonitis or dermatitis. And we then made or hypothesized that it could be possible to set up discovery pipeline to find novel tumor-associated antigens by seeing the overlap in mRNA expression of a tumor cell and the healthy cell, or if you want to study toxicity, the organ of toxicity, and then you can use the immune cells from the patient and then finally screen them. But I would like to bring you an example and not being too theoretical. So, for example, you can take here, you see the skin rash from this patient. You see here a dense T-cell infiltrate. This is a CD3 staining. You see the peritumoral stroma, and here the non-small cell lung cancer. Then you can isolate the DNA. You can take the PBMCs from the very same patient, do T-cell stimulations with the peptides pools spanning the protein of interest. For example, we looked here into keratin 6 or desmocollin 3. And you can already see here in this clonotype plot that the clone, which is recognized, that CD8 clone, which is recognized in keratin 6, is highly abundantly found in the skin toxicity, but it is also found in the lung cancer. Similar, we found that for desmocollin 3. However, you can also have some which can only be found in the skin, or some clones which can only be found in the lung cancer. For example, if it's a neoantigen, let's say it's a KRAS mutated tumor, and you would have a neoantigen specific T cell for KRAS, that of course would then only be found in the lung cancer. However, there's many shared clones in the skin rash and also in the lung cancer. In the very similar way, we also wondered what may cause the pneumonitis and what is the antigen which is relevant for the lung cancer. And there we found napsin A. Later on, again, I'm a dermatologist. I didn't know that napsin A is for non-small cell lung cancer one of the immunohistochemical markers. I was told that by a pathologist later, and indeed you can see it in the histology. But we were more interested into the T cells, which are specific for napsin, and indeed these T cells are more frequently found in patients who respond to checkpoint blockade in non-small cell lung cancer compared to patients who don't. And this was confirmed last year, I think, by a group from Seattle who took the same T cell epitopes we found for napsin and checked it in a larger cohort, and they could basically replicate and confirm this finding. But again, here it's the HLA-A*02:01 positive patients. And then I thought, well, this quite limits us. If we have always to match the HLA, it's a bit cumbersome. It's good, it works, but some people who have rare MHCs, they may not be able to be targeted with these soluble T cell receptors. And then I remembered I grew up studying immunology, or I could say I was taught immunology by mainly viruses, specifically VSV and LCV, two prototypic viruses, one cytotoxic, one not cytotoxic. And I learned there that usually it's not only T cells you have. And in cancer immunology, we had a very strong focus for a while on CD8 T cells because, of course, they're cytotoxic. They can delete the tumor cells. However, there's also CD4 T cells and there is antibodies, and you need those as well. And what we then in viruses to control an infection, and what we then did is we checked CD4 T cell responses for the same antigens, and we could find very strong CD4 T cell responses. And then we thought, well, what about B cells and antibodies in solid cancer? If we can identify such antibodies, they may be able then to be good new treatment modalities, and we will come to that in a moment. So first, we used the same cohort I have shown you already before, where we identified the keratin 6 or the desmocolin specific T cells with patients with skin rash and good response, or where we found patients with good response and napsin specific T cell responses. And then we measured the autoreactive antibodies against BP180. And maybe I should tell this audience that BP180 is a prototypic B cell antigen known in bullous pemphigoid. So this is a bullous disease which also occurs in patients without cancer. And it's well known because you can just run ELISAs for this BP180. And we could also see that lung cancer, so non-small cell lung cancer, also at least the squamous cell carcinoma also expresses this BP180. And we could see that patients who have this BP180 autoantibodies prior to treatment start with checkpoint blockade, these patients have a better overall survival. So somehow the autoantibodies also seem to play a role. And we then did analogous experiments for melanoma. It's not so well known autoreactive antibodies, so we had to develop that ourselves. This was a bit difficult in the beginning because, of course, you can just order proteins, but for example, the GP100 has a post-translational modification with glycans, and this you need to produce in eukaryotic cells. However, in this small cohort in 2019, we could already see that for some antigens, we could already see some hint that these antibodies may play a role. And we can therefore now conclude, before we move on, that both cellular and humoral immunity contribute to checkpoint inhibitor associated vitiligo. And then another paradigm change happened. So we are fortunate to live in these exciting times in how we treat our patients, because Patel and colleagues published in the New England Journal of Medicine 2023 this landmark story where they administered in patients with stage 3 melanoma with macroscopic lymph node metastasis. They either started checkpoint inhibitor treatment prior to surgery, so they already gave 3 cycles of pembrolizumab in the 3-week interval prior to the surgical resection, or they administered pembrolizumab for a year, just as it was proved at the time, in the adjuvant-only group. And what is striking is you use the same drug for the same time, but just in one setting, you do it prior to surgery, the probability of event-free survival is significantly better. And when we saw that, we thought, well, that's exactly our sweet spot. Because to study antibody responses, you should also, in virology, you should not do that when the host is full of virus, because then, you cannot trigger a very good and efficient antibody response. And we then started to set up in Tübingen, where we treat many patients using this protocol. We started to collect PBMCs and also serum from these patients undergoing neoadjuvant treatment for melanoma. And then also we took samples just at the time point of surgery. And we also took some of the lymph nodes and made single cell suspension so that we could also look into that. And what you can see is that patients with pathologic response, of course, you could also argue the architecture is destroyed here in this setting because you have the melanoma. But still, you can clearly conclude that the CD20 positive area and the CD21 positive area is quite different in a patient with a pathological response and in a patient without. Moreover, if we then did together with Thierry Nordman from Martinsried, some proteomics from this histo slides from these patients, we could see a B cell signature in the patient with pathological response, showing mainly variable heavy and light chain fragments present in the lymph nodes from the pathologic complete responders compared to the patients who did not show a complete response. Well, this is just association, but we then wanted to see if this signature translates also into an outcome of produced antibodies which are directed against the melanoma. And in the meantime, we also set up the production of our own proteins, which are recombinantly produced in melanoma cells, because we want to mimic also the exact post-translational modifications. And if we now compare the T0 to T1, so the serum levels of the antibodies prior to start of neoadjuvant checkpoint therapy at the time point of surgery, we could see a significant decrease of anti-GP100 antibodies in the patients who finally showed a pathologic complete response. And the same was true for another antigen for TRP2. And if you then look at the distant metastasis-free survival, you can see that patients who already had these antibody levels, higher antibody levels of GP100 and TRP2, they had a better distant metastasis-free survival. And we then thought, as I told you in the beginning, we would like to translate that in basically kind of from patients to patients, a new kind of drug development. So we set up a single cell B cell sorting by just coupling with AVITEC the recombinant protein with PE and APC, and then sorted into 96-well plates, sequenced the variable heavy and light chain, and made recombinant protein, recombinant antibodies, and coupled it with an antibody drug conjugate. And what you can then see here as a proof of concept you see here the UACC melanoma cell line, which expresses GP100 and TRP2. You see here clone A2 and here clone 12G. So they are derived from patients. And when they are formatted as antibody-drug conjugate, indeed, they can kill the tumor cells. If we take unrelated cell line MCF7, which is a breast cancer cell line, we don't see any effect of these antibodies. This already brings me to the end, to the future directions and take-home message. I would like to conclude that self-antigens are key drivers of both potent anti-tumor responses and checkpoint-related adverse events. Then emerging evidence suggests that B-cells response critically shape clinical outcomes under immune checkpoint blockade. This is also underpinned by these tertiary lymphoid structures which emerged in the last years. And shared antigen-specific T cells and antibodies may serve as a predictive mechanistic biomarker of both efficacy and immune-related toxicity. And finally, antibody-based therapeutic strategies offer a potential means to overcome HLA restriction and broaden treatment accessibility across diverse patient populations. And in contrast to adjuvant checkpoint inhibitor treatments, these treatments, of course, would not result in permanent damage to the patient, which is still sometimes a problem in our patients. And this already brings me to the end. I hope that I could convince you that autoimmunity is not always a bad thing. Of course, you have also the other side of the coin. Autoimmunity can also lead to a patient developing myocarditis, which can be lethal. And I would like to thank particularly my entire team here in Tübingen. So it's wonderful to see how the clinical part and the translational part can work hand in hand together. And I've also listed all the co-authors. Without them, it would have not been possible. And thank you for your attention. I'm happy to answer any questions.
Okay, thank you very much for this very insightful presentation. And already questions, please.
Thank you very much for the interesting talk. I have a question in regard of the generated antibodies. So if I understood it correctly, so these were all conjugated to a drug. So do you see also an effect of the antibodies alone? And based on that question, actually, do you think that the B-cell response could also be kind of a surrogate of a more effective T-cell response, or is it really rather a driver of the response?
Thank you very much for this excellent question. So to answer the first question, we of course wondered how these antibodies work. So they could either work using complement. So we ordered human serum, not complement inactivated, but which was made to study the complement response. We didn't see any effect of the complement. We then also did antibody-mediated cellular cytotoxicity, and to cut the long story short, there, particularly if you use NK cells, then we see a clear effect. In some antibodies, we also see an effect with granulocytes. However, we thought that it's more potent if we do antibody-drug conjugates. And as you are asking this question, I should also mention that we try to look into CAR-T cells to see if these antibodies are suitable to make CAR-T cells. And your second question was if this correlates with the CD4 but also CD8 T cell response. We are currently, for the entire cohort, we are doing all the stimulations with the T cells, and also we measure the ELISA antibody responses, and we are trying to correlate that and see how that fits together. That's a very good point, but I cannot give you the answer right now.
Okay, are there more questions in the room? Okay, so that is a question from my side. Is there anything actually, I'm an oncologist, so I'm not an expert in things like vitiligo, but is there any mechanism known how an inflammatory reaction could also induce this autoimmune disease? I mean, this could tell us something about how we could improve the efficacy of checkpoint inhibitors potentially.
You're addressing a question maybe in a broader sense, how can that be that we have autoreactive T cells and why a patient does not have vitiligo in the whole body, because we always think as a concept that autoreactivity is super dangerous because it will destroy the organ. What we think, but we have not clearly shown that yet, but we also know that from tebentafusp, so the T-cell engager, which is approved and which targets this GP100, it's never everywhere. So probably you need somehow an upregulation or a type 1 interferon stress in the healthy organ, which then results in the vitiligo or the toxicity. And if you ask the question, how could that so then be improved for checkpoint inhibitors? And we know that also from Christian Blank's recent publications that the interferon gamma signature in the tumor may tell us how hard we should hit and how strong we should risk autoimmunity.
Okay, thanks for this response. I just look into the room, but there's more questions, which does not seem to be the case. So thanks again very much for this very nice presentation. And we'll hand over to my co-chair, and then we'll look for the second part of this session.
Thank you. Our third speaker is Caroline Robert. She runs the melanoma service at Gustave Roussy and is also a professor of dermatology at Paris-Saclay University. And she will be talking about the benefits of combining existing checkpoint inhibitors and also novel checkpoint targets in melanoma.
Thank you very much. I'm going to give you a panorama of where we are, but mostly in melanoma, which is my field and which is quite at the forefront in solid tumor treatment. We are always showing this kind of slides with, the freeze, and we are so happy to see this improvement, which is true. As Lukas said, we have now 50% of our patients with metastatic disease alive after 10 years. It's much better than 15 years ago, where they were almost all dead after 1 year. So it's true, it's good. So you see the improvement. I'll come back to the different steps slightly after. But at the same time, we also have to admit that since 2015, with the combination of ipilimumab plus nivolumab, we are plateauing. So it's true that it's much better than before, but it does not go up at the same rate. And we need to find new targets or new therapy or new ways to give our therapies. And we should, the frustration is at the level of the expectations now. So when it does not work for one patient, it's really very difficult. So I will try to organize the talk like that, and I will be quite quick because I don't have a lot of time. But what about continuing with the best that we have, immune checkpoint inhibitor? I'll say a word about cellular therapy, although we'll have some talks about that later, so I won't spend too much time. Can we use still targeted therapy to improve and maybe change the way we give our treatments? So we have a new checkpoint blockade with LAG-3, which is authorized in melanoma, relatlimab plus nivolumab. It does better than nivolumab monotherapy in metastatic melanoma. There are some indirect elements to suggest that it would be as good as ipilimumab nivolumab. Personally, I don't think it's the case. The truth is that it improves the benefit-risk ratio because it mostly decreases the denominator, which is the risk, because we have much less adverse events than with the combination of ipilimumab nivolumab. So around 40% of grade 3 adverse events versus 60%. We have asked a lot of our colleagues in France and abroad to see if the combination of relatlimab plus nivolumab could overcome patients who had failed anti-PD-1 or combination with ipilimumab. And we found that indeed, there are some patients, about 30% of patients who can benefit, who can respond to relatlimab nivolumab even after failing previous PD-1-based treatment, especially if they had a secondary resistance. So it's worth trying if you don't have anything better. We now try to combine the 3 immune checkpoint blockers, CTLA-4, LAG-3, PD-1 blockade. Very few data, quite promising, less toxic. We use a lower dose of ipilimumab, but still too few data to make sure. And we have a new LAG-3 blockade with a Regeneron drug, fianlimab, combined with PD-1 blockade semiplimab, that gave really good results in phase 2 trial. We are going to have this year, at the end of the year, probably the result of a phase 3. I'm quite optimistic about this combination. I think it might be better than relatlimab. We have few data, but very interesting about combining immune checkpoint inhibition with an inhibition of the IL-6 receptor axis, with the double goal of decreasing the toxicity and also having an anti-tumor effect, because IL-6 has a pro-tumorigenic effect. And we have some good results if you compare with other trials with immune checkpoint inhibitor. Not enough patients to make sure that it's really great, but high response rate and much lower toxicity, even in this trial where you combine sarilumab with the three blockers. Now, we also use TILs for a long time now. It's like not a new treatment. You know that it has been authorized in the US with the company Iovance. We have also some T-cell engagers. We don't have CAR-T cells, unfortunately, in our melanoma and most solid tumor because we don't have the right target. What do we have? So TILs, on the left, there was this randomized trial, ipilimumab versus TILs in patients who have received PD-1 blockade, most of them. Very positive trial by our colleague from the Netherlands. Unfortunately, the control arm is not the right one because, of course, ipilimumab single agent is not something that we know we can do. Very low response rate and a few patients benefiting. And on the right, you have the Lifileucel trial, Iovance trial, that gave rise to the authorization. It was a phase 2 with about 30% of response in patients who had failed PD-1. They are running right now a phase 3 trial in first line, which I think it's absolutely not ethical from my personal point of view to give Lifileucel first line because it's highly toxic and patients can respond sometimes beautifully to immune checkpoint blockade. So I'm very happy because they opened a second line trial, again, the same thing as this one. And now they combine TILs with PD-1 blockade. And this is versus PD-1 blockade single agent, the phase 3. Now, we know that we have to improve when we inject TILs. We don't exactly know what we inject. So there are many ways to try to improve this strategy, to do some triage, to do some increase the fitness of these T cells. There are many teams that are actively working on that. One of the most attractive strategy has been presented several times already. We have very few patients, but I think that technology is so smart. It consists in transfecting the TILs with a regulatable membrane-bound IL-15. And this cassette is activated when the patients take Diamox, acetazolamide, which is totally not dangerous treatment. So instead of receiving high-dose IL-2, they take acetazolamide and it activates IL-15 bound receptor and it activates their TILs. So very few data in the patients, but I'm really looking forward to the following of this story. I would like to take the occasion to tell you a little bit what we are doing in the lab. We work for many years on how to modify the translation landscape, modifying the initiation factor of the translation, and we have shown that it can be effective in the context of targeted agent and also in the context of immunotherapy. And our ongoing work now is to confirm that when we act in a timely fashion on the regulation of the translation, we can actually take the lymphocytes that are very advanced in their exhaustion, terminally exhaustion pathway to put them backward. And we found that by using a modulator of this initiation of the translation, we can actually decrease the expression of a lot of marker of terminal exhaustion without affecting the proliferation of T cells, without affecting their IL-2 production, their interferon gamma production. And when we do co-culture of autologous lymphocytes, melanoma cells, or when we do organotypic experiments where we put fresh slices of melanoma and we combine modulator of EIA4F, more precisely EIA4A, one of the compound of this complex, together with anti-PD-1, we actually increase the tumor cell lysis and infiltration of T cells. It works beautifully well in several mice models, like syngeneic melanoma or colon model, and you can see the sort of vitiligo of the tail of the mice when they respond very well. But, and in parallel, we can see that the molecules that are associated with the most terminally exhausted profile, they're actually decreasing when we combine these drugs, and we have an increase of the progenitor cells that we know are more likely to respond to PD-1 blockade. Now, coming back to what I was telling you before, we wanted to see if this could be used ex vivo to treat TILs before we re-injected to the mice. So we prepare TILs as we do for patients, and then we inject the TILs. Actually, we prepare the TILs from patients and we re-inject them in immunosuppressed mice where we have implanted PDX from the same patient. And we found that we have actually an increase of the efficacy of these TILs. It's not so easy to do TILs in mice, actually, but it works well, and we have a decrease of the proliferation of the tumors when we modify by modifying the translation profile of these T cells. So coming back to what is actually, because this is not in development yet, maybe hopefully it will be, but coming back to what we have right now, ImmTAC. So Lukas told us a little bit about that. As you probably know, tebentafusp, so mobilizing T cells against GP100 expressing melanoma cells in the context of HLA-A2 gives good response in terms of overall survival in uveal melanoma. It was the first drug approved showing a benefit in survival, although the response rate is ridiculous. It's very low. They respond very poorly, but they live longer. It kind of seems to slow down the proliferation of the disease. And interestingly, it's very well correlated with the ctDNA decrease, which is not so rarely seen, this good correlation. And the scientists at Immunocore, the company who developed tebentafusp, they found a sort of fitness signature, which it can be found in the blood. T cells, when they have high stem cell signature and effector signature, they seem to be associated with a good response. So I think it's an easy marker that we could easily look at because it's in the blood at baseline. Maybe we should look at that for other treatments too. So tebentafusp now, in combination with PD-1 blockade, is currently tested for patients who relapse after PD-1 blockade. Trial is ongoing, it's recruiting well. And they have another ImmTAC that targets not a GP1 peptide, but a PRAME peptide, still in the context of HLA-A2. First line, not easy to enroll because versus PD-1 monotherapy, but we'll see. These are the results of the phase 2. Now, something that is quite attractive because of the result. You see the waterfall graph of the phase 1-2 with uveal and cutaneous melanoma post PD-1. This is a genetic modification of leukocytes that are extracted from the blood. So they express again the TCR that recognize the PRAME peptide, still HLA-A2 restricted, but I mean, the results are quite striking, although we don't have a lot of patients, and there is an ongoing phase 3 who are participating to this trial, and we are quite, impressed. Another treatment that surprisingly gives close to 30% of response in a multi-cohort phase 2 trial. You see, it's about 30%, including patients who have progressed after ipi-nivo, which is the best that we have today, is the combination of PD-1 blockade plus an oncolytic virus derived from herpes that is injected intratumorally. Phase 3 is running now, quite courageous because it's the only phase 3 that is only post-ipi-nivo, and it's versus whatever you can give. There is not much choice. But if it's true, it's very interesting because it's absolutely not toxic. It has almost the same toxicity as PD-1 single agent. So that makes a big difference because all of the other TCR-based therapy, they're quite toxic. Especially the ones that require chemotherapy before, like TILS or the hematic trials, quite severe chemotherapy before. Vaccines. So, I don't have to tell this assembly that we know how to design neoantigen vaccines now. It's a quite complex process. We also know that we have neoantigen in the normal existing patients, but they are very rare. Their representation is very low. They are present at 0.000% compared to other antigen that you can see. So we can do it. Merck and Moderna are able to provide you with an RNA encoding neopeptides after you provide them with a sample of tumor, like paraffin-embedded tumor. And that was quite a tour de force technology because they can give you the RNA vaccine after about 6 weeks. And there was this trial, phase 2 trial, adjuvant, after the resection of a stage 3 melanoma, and you treat with anti-PD-1 with or without this RNA vaccine that is totally personalized. So usually the vaccine arrives a little bit after you initiate your PD-1 treatment, and this is the standard adjuvant treatment versus adjuvant PD-1 plus vaccine, giving very good results. Phase 2, so it had to be confirmed by a phase 3 that has been done very rapidly. We have enrolled the patient very rapidly. Patients who are very happy to enter into this trial will have the results in the coming months, I guess, or 1 year. But I'm not the only one to wonder whether it's really the right things to do. Are we really targeting what needs to be targeted? You all saw this paper, for example, with a COVID vaccine, like patients receiving immune checkpoint inhibitor for melanoma or for lung cancer. When they have received the COVID vaccine, 100 days before or after, they seem to do better. And it doesn't seem to be because the other one died of COVID, because when they do the experiment in mice, there's quite convincing data that they activate the innate immunity, interferon alpha pathway, and that it's actually working well. So, maybe it could be effective, and would it be even as effective as this very time-consuming neoantigen vaccine? I don't know. I'm asking the question. Then there is this very nice work from the team of Claude Perrault in Montreal, where they found that unmutated, so-called aberrantly expressed tumor-associated antigens seem really to be the ones that do the job compared to mutated antigen. And in fact, there are very few mutated sequences that seem to be responsible for the response, and they used melanoma and lung cohorts. And rereading some other work, by the paper of Reyes and al., they also found that the clones, the tumor clone that decrease in responders, they are the one that actually are encoded by non-mutated antigen, and very often dark, also from the dark genome, like intronic region, repeat region. So maybe we should try more to look into the hidden face of the iceberg instead of always running after the thing that we see. And when we see them, in fact, we see these tumors in the tumor, but the tumor are growing. So it means that these cells are maybe not the ones that are the most important. Then you also saw all this data, retrospective data, about the fact that when you inject your immune checkpoint inhibitor early during the day, it's better than when you inject it later. There is now a prospective study that has been presented at the ASCO and published. It has some pitfall in the design, but still, I think it's quite interesting, especially probably for the first injection. And there are some hypotheses that are holding on, I think. Plus, if you remember the data on mice, you know that mice, that has been well proven that when you vaccinate them during the night, it works better. Mice are nocturnal animal, unlike us. So maybe it could work also for us that it's during the day. And also I'm not going to talk about fecal microbiota, you know all the data about that. Still a lot of uncertainties. We have a clinical trial that is going to be, we are going to have the results very soon with a randomization, PD-1 blockade, CTLA-4 blockade, together with or without fecal transplantation randomization. I'm really looking forward to have these results. So just to say that, okay, we're happy, we're treating some patients, but we should really not sleep on our laurels. And I especially say that to pharma company, because sometimes they tell us, okay, for melanoma, it's done. Now we're going to be interested in other tumors, which I don't think is a good idea. And of course, I would like to thank all the people who work in the lab, in the clinic with me, and our sponsors. Thank you very much.
For a very, very comprehensive overview of the field and also for interpreting the data for us.
We have questions in the back, please.
Hello, is this working?
Yes.
Perfect, thanks. Thank you for the nice talk. I just had a question about this EIF4A inhibition. So you showed that, and correct me if I'm wrong, but you said that when you inhibit this translation initiation factor, you get increased cell proliferation. I was just curious how that works exactly, because I've been under the impression that when you inhibit translation, then you get decreased cell proliferation. And so I'm curious, like, if what is happening at the translational level there when you do this inhibition?
Yes, that's a good question, and that's a question we always have when you act on these factors. First, you have different types of EIF4A inhibitor or modulator, and they act differently according to the 5' UTR of the RNA that you are considering. And it's not an all or nothing effect. It's not like you decrease all the translation. Plus, sometimes you decrease the translation of something and then you increase the transcription. I mean, you have indirect effect. And it is true that the idea that we have when we use these modulators is that we want to fight against an adaptation of both the tumor cells and the immune system to the stress of the treatment, in fact. And we have seen, from the tumor side and the immune side, that this is a very important way of adapting to the stress that is induced. And in fact, when you use the same EIF4A inhibitor for tumor cells and immune cells, you absolutely don't have the same effect. Same thing if you use the same drug on, for example, tumor persister cells who have seen the treatment and the cells that have not seen the treatment, you don't have the same profile of inhibition. also. So it's very depending on the state of the cells and on the complexity of the 5' UTR sequence. And in fact, on T cells, it does not impair their proliferation. It's even increase it a little bit, and it modifies, I mean, a lot of expression. I showed some of them, but we have done an extensive RNA-seq, and we mostly decrease the expression of the markers that are involved in exhaustion. I mean, activation, but then exhaustion, whereas it does not decrease, for example, interferon pathway and things like that.
Thank you. Further questions to Caroline? I'm glad you brought up the FMT in one of your very last slides. Is there a role for FMT in boosting checkpoint blockade in cutaneous melanoma and beyond, in your opinion?
Well, you know, the data, they are interesting until now. Most of them, except one trial in renal cancer, they are mostly single arm. And for example, the very interesting data by Bertrand Routy and Ariel Elkriev that were published in Nature Medicine, it's on melanoma, PD-1 blockade plus or minus CTLA-4 with fecal transplantation. It gives high level of responses, but mono arm, and plus the population was quite, not very much patient with ILDH, sort of good population of patients. So I don't know exactly what is the effect. I think also we have to make sure that we don't damage the microbiome, because we know more and more that some people have good, maybe some people have a bad one. And if you transfect something to somebody who has a perfect microbiota, maybe it's not good. So we probably should check it before. And we would like to know better what is good and what is bad.
Good. Thank you, Caroline, for a lovely presentation again. Now we move on to the flash speakers. We have the first speaker is Surender from the University of Tübingen. You're a PhD student in the lab of Professor Tabatabai, whom some of us still know because she used to work here a long time ago. And we look forward to your presentation.
Hi, good morning, everybody. I am Suri. You can call me Suri, by the way. And right now I'm finishing my PhD in Tabatabai lab in Tübingen, and I'm happy to be here today. To give you a brief background about our scientific hypothesis, glioblastomas harbor RB1 pathway alterations that supports inhibiting CDK4/6. However, the clinical benefit has been very limited as being shown in the recent clinical trials. Preclinical data suggests that abemaciclib, which is the CDK4/6 inhibitor, it enhances anti-tumor immunity, which led us to hypothesize that using this inhibition may prime these tumors to immune checkpoint blockade. At first, by using immune peptidome analysis, we show upregulation of both MHC class 1 and class 2 peptides and glioma cells under abemaciclib treatment. And then most of these peptides are either interferon alpha or interferon gamma response-related pathways. That shows that this treatment leads to immunogenic response in glioma cells. Next, we test a combination of abemaciclib and anti-PD-1 in 2 glioma mouse models, where we show that combination treatment prolongs survival significantly and also leads to infiltration of CD4 positive and CD8 positive T cells. Next, we wanted to understand whether T cells are the mediators behind this response, which is related to enhanced survival benefit. We used a T cell depleted mouse model where we showed that the combination treatment did not have the same drastic effect. And then by using neutralizing antibody experiments, we showed that inhibiting CD8 positive T cells had the strongest negative impact on survival benefit, which indicates that these may be the drivers behind this strong phenotype. To summarize, abemaciclib, which is a CDK4/6 inhibitor, it induces immunogenicity in glioma models. It synergizes with anti-PD-1 to prolong survival, and it appears to act through CD8 positive cytotoxic T cells to promote the survival benefit. Thank you very much for this, and I'm open to any questions.
I'm afraid we really don't have time. Maybe one short.
Nice.
there have been a couple of papers showing the CDK4/6 inhibitors impact CD8 differentiation. Did you look into that?
Yes, I did. In fact, there is this brain metastasis breast cancer model where they also showed the same. And in general, they always dive into the CD8 compartment, but they usually don't see what mechanistic pathways are involved in there. So we are also trying to answer that. We are using ultra-low input RNA sequencing to further advance that and find therapeutic targets. Yeah.
Great. We move on to the second flash speaker. André, Rosa, that is you. André will be talking about cancer-on-chip models.
Well, thank you for the opportunity to present our work on organ-on-chip models. Let's talk about our cancer-on-chip system that allows to assess efficacy and safety of CAR-T cell therapies against solid tumors. Now, about this specific organ-on-chip, this is made of 6 sequential chambers where you can load cancer aggregates separated in a tumor chamber separated from a media channel by a porous membrane that can be endothelialized. Now, with this microstructure, we can perfuse immune cells. And for this specific case study, we're looking into ROR1 CAR-T cells that were engineered to target breast cancer tumors. Now, with this approach, we could analyze and quantify the immigration and perfusion into the tumor aggregates from immune cells. We look into the parental T cells, but also ROR1 CAR-T cells, in parallel with the analysis of tumor growth, kinetics, and also the analysis of tumor cells. This system was also, of course, amenable to the use of fibroblast aggregates to assess off-target effects. Looking at the effluents, what comes out of the chips, we could measure or recapitulate clinical relevant cytokine levels, specifically for CRS. This was in chips that had tumor aggregates alone, but also with macrophages integrated at the same time. And finally, we could pharmacologically modulate the cytokine release, in this case using dasatinib, using different regimes just to look into the prevention of cytokine release, but also the regulation in this in vitro system. Finally, I just wanted to highlight our latest publication where we use this exact system as well, which has a broad potential, where we look at the specificity of a new nanovaccine targeting pancreatic ductal adenocarcinomas. With that, I just want to thank for your attention, and if you have any questions.
Truly an exciting model. Any questions? Short one? If not, let's thank again all speakers of the session for inspiring presentations.
Hey, it's my pleasure to introduce our next speaker, who is Peter Elmark, and he's from the University of Lund and also from Alligator Bioscience. And he's going to talk about reprogramming tumor-associated macrophages.
Thank you. Thanks for the introduction, and thanks to the organizers for inviting me. Is that better? So I'm going to talk about reprogramming macrophages and other myeloid cells, and this will be in the context of reprogramming them through the CD40 receptor. So I work at a company called Alligator Bioscience, and we developed, among other things, a CD40 agonistic antibody, and that's going to be the focus of the talk today. I'm also working at the university doing translational work, but the focus today will be on the work that we have done with the CD40 agonistic antibody called mitazalimab. So first of all, why are we targeting myeloid cells? Myeloid cells are, as you know, they are quite abundant in many tumors. They play a key role in regulating the tumor microenvironment, and they play a key role in the cancer immunity cycle, and they are important for getting what we all are after, that is more T cells that can recognize and kill tumor cells. There are a number of therapeutic strategies being pursued when it comes to targeting myeloid cells, inhibition, depletion, repolarization, or reprogramming, and that is really what CD40 targeting is all about. So CD40 is a receptor expressed on many of the antigen-presenting cells, myeloid cells, such as dendritic cells, and if you stimulate CD40 on dendritic cells, you can enhance uptake and cross-priming of tumor-specific antigens to T cells, allowing the dendritic cells to license the T cells to then go and kill tumor cells. CD40 is also expressed on tumor-associated macrophages, and when you stimulate CD40 on these macrophages, they get more sort of shifts from an M2 to an M1 phenotype, if I dare to speak that type of language here. And in the end, that is known and it's been shown, and we have some data to support that here as well, that this can lead to the immune environment being more permissive and also lead to the degradation of stroma. So the antibody that we have developed, and there's been a number of CD40 antibodies evaluated also in the clinical setting over the years, and mitazalimab is quite straightforward antibody. It has a wild-type IgG1. It binds to domain 1 on CD40, and it's sort of the best-in-class properties is down to the epitope, which provides strong agonistic activity, but it also provides Fc gamma receptor engagement or conditional agonistic activity. That means that it only activates CD40 expressing cells when it binds to CD40 and at the same time engage Fc gamma receptors. This makes, of course, the Fc important, and there's been sort of a lot of trials, I would say, in the clinical settings of different Fc gamma receptors being employed. I think that I'm not going to go into exactly why I think that is the case, but I think the data we have really shows that the wild-type IgG1 provides the right balance of immune activation. And that is important because what we can show is that with our antibody, we do get immune activation, but we don't get any added toxicity, and no cytokine release syndrome has been reported in our studies. And I think that is key to allow for efficient combination therapy. We have shown that this holds up in phase 2 study in pancreatic cancer patients, where and this I'm going to talk more about this study, so I'm not going to go into detail here, but sort of the efficacy and tolerability in that study is really what we are makes us want to pursue this antibody further. There's also opportunities in other indications beyond pancreatic cancer. We are sort of the key things we are working on now is to get the phase 3 study going. That is sort of a challenge in itself. But in addition, we have a number of investigator-initiated trials ongoing or about to start, which is quite exciting, where we combine CD40 also with other agents such as checkpoint inhibitors and other immune modulators. And just so in addition to the monospecific antibody, we have a bispecific CD40 antibody that is, I found very interesting, but I'm not going to talk about that today. So we know that CD40 antibodies, and mitazalimab is no exception, it synergizes with chemotherapy, both in anti-tumor activity, but also in the ability to reduce M2 type macrophages and increase the number of cytotoxic T cells in the tumors. We know that if you take tumor-associated macrophages and stimulate them in vivo, so this is patient-derived macrophages, or you get a clear activation of these macrophages. And this is really what led us into going for pancreatic cancer, which is an indication we do have a lot of infiltration of macrophages. So pancreatic cancer is one of the leading cause of death in the Western world. It's a quite was a very aggressive tumor type. For most patients, they are presented with metastatic disease, where the only option is to get chemotherapy. There are broadly today, there are two main chemotherapy regimens: the Gemnab combination or modified FOLFIRINOX or sort of similar derivatives. That being said, there's not much happening in PDAC over the last decade or so in terms of improving overall survival. That will change, I think, over the next couple of years. It's not going to only be mitazalimab, but there are also a lot of KRAS inhibitor data coming out, and then the field will actually move forward. I'm quite convinced of that. But coming back to why mitazalimab works, well, we have it's a tumor with a dense stroma, highly infiltrated with suppressive macrophages, no T cells, and the checkpoint inhibitors shows no activity. So when we designed this study called Optimize-1 in first-line metastatic cancer patients, we took some care to design the study and sort of differentiate the study towards what's been done before. So when you look at what we did was that we have a dosing regimen where we have a lead-in administration of mitazalimab. So the first thing the patient sees is a dose of mitazalimab, enabling activation of tumor macrophages and enabling sort of a sensitization of the tumor before the chemotherapy is given. And in all subsequent cycles, the patients get chemotherapy and then mitazalimab 2 days after. But the first cycle is really geared towards maximizing the activity of the CD40 antibody. And this is how it looks like if you look at the dosing regimen. So you can see that the cycle 1 is really what stands out, and then you have chemo and mitazalimab given 2 days after. The anti-tumor activity that we could measure in this study was, so we got very deep responses, as you can see in the waterfall plot. We had, and I would say also the primary endpoint of this is a single-arm study was overall response rate. We met the overall response rate criteria. The study was positive. But I think what's important here in pancreatic cancer is always going to be overall survival and the duration of response. And what you can see also in the spider plot is that we don't only get deep responses, but these responses were quite durable. And we have patients that has been on study past 3 years, and in several cases on mitazalimab only for the last several years. When we look at the overall survival from the study, so looking at the median overall survival, it's 14.9. That can be compared to 11.1 with Prodigy and Napoli-1. So improving survival by 3.5 to 4 months. But I think the important part and what we really are excited about is the latter part of the curve, because we see some really unprecedented numbers on the 24-month and the 30-month overall survival. So this is a single-arm study. We have, so to substantiate that the findings we are seeing is relevant, we have also done a literature-based indirect treatment comparison where we can show that we are significantly better than the pooled data from other published studies. This was done before we had the final readout, so you don't see the full survival curve in this comparison. So to sum up the response data, the median overall survival is 40.9 compared to 11.1 in historical controls. I think the impressive part in the data is the duration of response. So if you get the response, it's really durable. And if you look at the long-term or the 24-month overall survival rate, it's quite unprecedented compared to controls. And we are we are, based on this and other data, we are preparing now for a phase 3 study. Should also mention that the dose we have here is 900 microgram per kilo, so it's the dose that we have agreed with FDA for the phase 3 study. I don't have the data here, but we also had to amend this study to satisfy or sort of to address the Optimus Project Optimus initiative by the FDA, which was quite sort of time consuming, but it also provided some additional data that I think is going to be helpful to further understand the molecule. I don't have the data here, but what we could see is that the 900 microgram dose, when we did the exposure response comparison, was actually superior to the 450 dose that we also tested. So further substantiating the anti-tumor activity we see in Optimize-1. Now, I'm going to switch into the translational part of the data. So what we aim to do, as I said earlier, is to induce stroma degradation, sensitizing the tumor to treatment, reduce the immune suppression, and then have more activated T cells. So looking at what happens on the cytokine level, we can see that, and this is what we expected, so we get a rapid upregulation of MCP1 and other myeloid sort of associated chemokines, cytokines. We get the delayed expansion or induction of interferon gamma and IP-10. We could also see that we activate circulating antigen-presenting cells that express CD40. So what we see is, if you look at the top row, you can see that you have an early activation of B cells and monocytes, and then you have a sort of a delay to cycle or to 1 week after treatment. You can see an expansion of proliferating CD8 or T cells and NK cells. You also see in the bottom row that you do have an activation-induced reduction of circulating immune cells, B cells, actually also T cells, monocytes. You can see that, and that is what we expected. But the assumption is that they are activate. It's activation-induced migration to tissue tumor. Looking into what happens in the tumors, we had from this study we had 3 paired biopsies that we were able to analyze. These were all from patients with partial response, so that was the upside. We were always disappointed to see the number of the low number of biopsies, paired biopsies we can get on treatment, but that is sort of what it is in pancreatic cancer. But what we could see from these 3 samples is that we see the expected upregulation of genes associated to myeloid and T cell activation in the tumor microenvironment, and downregulation of expected genes. So while a small and limited data set, we can see that the data from treated tumors point that we do get the type of activity that we anticipate to get. So the design of the study actually allowed for looking at CD40 only activity and how that relates to outcomes. So this is actually the only study that I know of that are combined with chemo that actually have sort of the opportunity to look at what happens after the first dose of CD40 before you get any chemo and how that relates to clinical outcomes. What we could see is that we had a large number of T cell population, KSH7 positive T cell, NK T cell, and NK cell populations that were associated with better prognosis. So activation before any chemotherapy is actually associated with better survival. We also looked at the baseline biopsies from these patients and looked for association. This was done by RNA-seq and gene expression analysis. And what we can see is that we found pathways related to extracellular matrix organization that were associated with response, and we sort of look at these as targets for CD40. So we were quite intrigued in seeing this, and it's also something that matches up well with what we see in preclinical data in mouse models. This is also substantiated with data from another study that was done. So this is an IIT that was published last year, but I wanted to bring some of that data because it shows it also in a limited set of patients, but it shows that you do get that type of activity on the collagen in the tumor, then relating to an sort of increase in CD3 T cells, further substantiating that we do enable activation of macrophages and the restructuring of the stroma. So I'm gonna finish up here, and I hope sort of the data we've shown here can illustrate how by targeting CD40 with a safe and efficacious CD40 agonist can lead to activation of immune cells in the periphery that can migrate to the tumor area. We can induce immune activation into a microenvironment that sensitizes the tumor, and that this in turn leads to a prolonged survival and duration or a durable response. And I think when I look at the data, I think the biomarker data strongly supports that the survival benefits that we see over historical control is really down to the mitazalimab treatments, and I'm really looking forward to seeing this translating into a phase 3 study. And I would like to thank all of you for your attention, but also the families, investigators, the Alligator teams, and the collaborators. And I'm happy to take any questions. Thanks.
Thank you very much for this very nice talk. Questions? Yeah, Kara.
Yes, thank you very much for the presentation. Quite amazing data. I'm thinking about if you have a study perform correlative study to understand the patient working better than the other, and particularly if you ever checked the ratio between myeloid cells and lymphocytes before infusion, if this needs to be balanced, if both are involved, or...
Yeah, I mean, the neutrophil to lymphocyte ratio is important. So that's one, but that's a known sort of, that is known for pancreatic cancer and chemotherapy overall. And so what I've been looking for, and still look for, is really to find a good and solid immune status biomarker that can explain and find the right patients. I think we have some clues based on this data on how to select patients, but I would say that the phase 3 study will be sort of not a biomarker-rich study. We don't have enough data to support that at the moment, but I think we need to get there, and I'm still working on getting there.
Yep, briefly, I guess maybe I just didn't follow this part where you were talking about the engagement of tumor-associated macrophages. CD40 is expressed by B cells, dendritic cells, and what have you. And the response that you see is very strongly indicative of a DC and B cell response. So do you know whether you actually get into tumor-associated macrophages within the tumor microenvironment, whether your antibody binds and engages these cells specifically at all?
So, yeah, great question. And I think I don't have any data to show that you actually get to the tumor-associated macrophages. So the data we have from this study is really it's RNA-seq bulk data, and it's not enough to really answer that question specifically. I do suspect that sort of the response we see, I think it's going to be dependent on activation of both macrophages and dendritic cells, but I don't have the data to sort of formally prove it from the study.
Mm-hmm. Next question.
Very nice talk, congratulations. My question is similar, which is, you said that Fc engagement via the wild type IgG1 is very important. And so how much do you think it's actually activation of macrophages or dendritic cells via the CD40 agonist? And how much is it possibly just killing of myeloid suppressor cells and macrophages in the tumor microenvironment, which are often very suppressed?
Yeah, that's a good question. So while the FC is a wild type IgG1, I think from our at least from our preclinical studies, we see no real role in any depletion whatsoever. And I think if I wanted to deplete myeloid cells, it's not the wild type IgG1 CD40 antibody that we use. Yeah, that's the short answer, but it's a good question.
Mm-hmm. Sonia?
I would have a follow-up question. So first, thank you very much for the talk. It's really impressive. And regarding also this one type IgG1 design, I was really wondering how do you avoid the sink effect in the liver? So I would expect some type of cytotoxicity maybe from the Kupffer cells, or is it that your chemotherapy is somehow remodeling the vasculature for intratumoral delivery? Sorry.
Yeah, so I don't think that the chemotherapy is involved in this at all. We can see that the PK is unaffected by the chemo. That was one of the questions I really had, because we have the data here to show that we have PK both before and after chemo. So we don't see any difference in exposure based on the chemo. We do see, the CD40 is one of these targets. We have a very strong target-mediated drug disposition. So what we can say, and this is very similar to many other CD40 antibodies, that we have a half-life of about 24 hours. We have a receptor occupancy over time that is much longer. So the cells in the circulation are occupied for much longer. And then the question is, and this is something also I've discussed a lot with a lot of smarter people than me, is really what type of receptor occupancy would you need over time? It's you want an agonistic activity, and then there's always a risk of exhaustion and so on. I think the data we have suggests that we get at least based on the clinical data, that we get the sufficient receptor occupancy with the dosing regimen we have here. We haven't tested everything, of course.
Mm-hmm. Thank you very much again.
Thank you.
So then we have two quick flash talks before lunch. And the first one is from Sheila Plaza from Philochem.
Thank you.
And you're on.
Okay, thank you. Thank you for the kind invitation and welcome to my flash talk. I will present a combinatorial approach to enhance tolerability of immunocytokines. And immunocytokines have been the main focus of our group, and basically they fuse a cytokine payload to a tumor-homing antibody, allowing long-term tumor accumulation of the product within the tumor. However, they suffer from those limiting toxicity, and patients may experience adverse events shortly after infusion. And this is correlated to basically peak cytokine blood levels that can induce on target of tumor activity. So new ways to improve tolerability are needed, and we have developed the intracork technology, which relies on the combination with signaling inhibitors. with a combination of signaling inhibitors that can mask activity of the cytokine in circulation when the levels are very high. And the use of small molecules, it's ideal because they are normally clear from circulation very rapidly. Therefore, the long-term anti-tumor activity will not be impacted. This idea has been preclinically validated, and here you have an example with a tumor-targeted IL-12 product that in combination with the cork allowed the mice to receive high doses of the IL-12 fusions, preventing body weight loss. Importantly, anti-tumor activity was maintained, confirming our initial hypothesis. And this strategy is a versatile tool that we have published with other immunocytokine products, and we are now in our way to transition this idea into the clinic, and I hope I can bring more data in the near future. Thank you for your attention.
Thank you. Questions? Yeah.
Hi, Caroline.
I'm not sure I understand. So you give at the same time the JAK inhibitor?
Yes, exactly. So, I mean, you need, this is a key point. Basically, you need to kinetically match both components because you need to understand what is the toxicity profile of your product and then try to apply the antidote in order to match when it's peaking high, it's peaking in the blood.
You don't give long-term JAK inhibitor.
No, you don't want to do that. Also to reassure that you don't apply extra toxicity with the JAK inhibitor. Yes.
Might not be so good.
No, exactly.
Thank you.
Yeah, thanks to you.
Okay, thank you.
Thank you.
And the last one is from Michael Schmidt from Vir Biotechnology in Bellinzona.
Hello, everybody. And I would like to talk to you about T-cell engagers. And in the last years, it has really emerged that they can be very potent, but there's a big problem that even if just small amounts of the tumor-associated antigen are expressed in healthy tissues, you can have high toxicity there. Our approach is to have a double mask, which masks as well the anti-CD3 part, as well as the anti-tumor targeting part in small T-cell engagers. And what this mask actually looks like is this globular structure, so really double masking both sides. The other big advantage is that this increased molecular size really increases the half-life in circulation. Then at these yellow spots on both sides, you have cleavage in the tumors via tumor-associated proteases like MMP9. That activates the T-cell engager, but at the same time, it makes the molecule much smaller and decreases the half-life in case any of that active TCE leaks out of the tumor. So further increasing that half-life, short half-life increases the safety again. This is an example about one of the three molecules we have in phase 1 clinical trials right now, which is against HER2 and breast cancer or colorectal. And you see that in vitro, on the left side, there's a more than 10,000-fold difference between the unmasked TCE and potency. There's no proteases present compared to the masked ones. Instead, in vivo, in the top, near to the vehicle, no much effect if there's an uncleavable linkers. But if you have cleavable linkers, you have the same potency because of the cleavage in the tumors between the unmasked and the cleavable masks. Similarly, we have two other molecules, one against EGF receptor in the clinic and the other one against PSMA. The last one against prostate cancer, please check out our homepage. You'll find the presentation there. It seems to be very spectacular and also very low toxicity results in this phase 1. And as a last slide, I want to just say in Bellinzona, we are focusing on the discovery of these new antibodies parts, engaging the tumors. And we have a lot of years of experience on infectious disease, of course, but now we translate that into tumor targeting and then incorporate these binders into the T-cell engagers. And so currently, and this is also what my team is doing, and we're currently working on 7 new preclinical models. So there's much more to come. Thank you for your attention.
Thank you. Yes.
But you have to rely on the presence of these proteases, and is it always the case? I mean, it may not be the case for all tumors and all metastases. And all.
Yes, this is very true. One good tendency is that metastases seem to have even higher expression of proteases. And the other is that we are also using that, of course, to select which indication we go for. Because we have what indication is expressing a certain tumor antigen, associated antigen, and then also we're checking for profiling of those proteases to then have the best match. But it's a very good question.
Okay, thank you again. And also thanks again to all the speakers. And there's a lunch break now until 10 to 1, I believe. And then all the speakers should come down to take the pictures first. And Goethe.
Okay, so we switch. With this, it's a great pleasure to introduce the next speaker. So Magnus Essand is a professor at Uppsala University in Sweden. And beside different vector systems, he's working also on next generation CAR-T cells, translating this from preclinical research to patients. And this is what your talk is about. Looking forward to your talk.
Thank you so much, Tobias, and thank you every organizer of this conference for the invitation. I will speak about optimizing CAR-T cells, mainly to improve their efficacy, but also a little bit to reduce toxicity. When it comes to arming, I have a disclosure. Me and my colleague DU have started a company, so the IPs for this arming is in this company. As an introduction, I would like to say that we were early with CAR-T cells in Uppsala. I believe that we treated the first patient in Europe, actually, back in May 2014. And that was thanks to my colleague Angelica Luskog, having done a postdoc with Malcolm Brenner and Jan Pietro Dotti at Baylor College of Medicine, and brought the technology back to Sweden. And when she was back, we applied for a grant, and we could produce a retrovirus batch at Malcolm's lab and start a clinical trial. So we have produced two clinical trials with 15 and 24 patients in the past. Today, of course, the landscape has changed. We have a lot of approved treatment, 6 or 7 products being approved for B-cell malignancies and plasma cell malignancies. CD19 CAR-T cells for leukemia and lymphoma, and BCMA CAR-T cells for multiple myeloma. So there are still challenges. In some cases, patients are completely refractory to the disease, and then there's probably something to do with the intrinsic properties of the tumor cell or the CAR-T cells. We have early relapses, which is very often related to poor T-cell engraftment. And when we have later relapses, it can also be due to engraftment, of course, but it can also be related to antigen loss and antigen escape, so that the tumor are downregulating the antigen that is targeted by the CAR-T cells. We have toxicities, of course, CRS and ICANS when it comes to CD19 CAR-T cell therapy. And we have the unmet need when it comes to solid tumors and CNS tumors. And there, the problem is immunosuppression into microenvironment and also antigen heterogeneity. So antigen heterogeneity is kind of mimicking antigen loss in CD19 CAR-T cells, meaning that you lose the target on the tumor. So I will focus a little bit on this and how this can be addressed. Oops. So it's also shown here. With leukemias and lymphoma, you can end up with CD19 negative tumor coming back. So there can be a downregulation or a rearrangement of the protein, so it's no longer on the surface of the cancer cell. When we look at solid tumors in the bottom here, we have heterogeneity already from the start, and that means that targeting a single antigen is very likely going to lead to development of a clone that is no longer responding to the therapy. So we need to address that. One can use, of course, one can target more than one antigen using tandem CARs, or T cells with multiple CARs, or even multiple CAR-T cells to address that. Or one can arm the CAR-T cells, and it's quite popular nowadays, and often cytokines are being used, IL-12, IL-15, IL-18, all show great promise. Carl June presented data on IL-18 armed CD19 CAR-T cells, showing that he can have responses in patients that have relapsed after conventional CD19 CAR-T cells. So just by adding IL-18 as a cytokine that secreted from those CAR-T cells, he can get responses again. We can of course produce bites on nanobodies, and Marcella Maus' example of that, targeting the truncated EGF receptor, the EGF-B3 in glioblastoma, and then having a secretion of bispecific antibodies targeting the EGF receptor. So once you are in the glioblastoma environment, it's safe to kind of release those antibodies, and you can then target EGF receptor as such and linking it to PD3. So there are good examples of arming strategies. We have chosen to look a little bit, take it a little bit differently. We are focusing on activating bystander immunity and epitope spread, and have chosen to arm CAR-T cells with a bacterial-derived factor called NAP for neutrophil activating protein. It is derived from Helicobacter pylori. NAP is a little bit misleading. It is actually chemotactic for all innate immune cells, and even for B cells, and it has very strong Th1-inducing properties. So we have used it. So we have used it in the past for arming oncolytic viruses. So we have a lot of knowledge what NAP can do when it is being released in the tumor microenvironment. So induce IL-12 and IL-23, for example, but also activate dendritic cells and induce their migration to a draining lymph node. So it took a while before we decided to arm CAR-T cells with NAP. It's one thing to put it in an oncolytic virus, but now we're putting a foreign protein in our CAR-T cells, meaning that they can also be targeted for destruction if NAP peptide is presented by MHC. So we debated back and forth before we tested, but we have tested that. And this is also to show a little bit about arming. So often, if you go from unarmed CAR-T cells to arming, you can increase efficacy, but often also toxicity is being come along with this, and this is especially true for IL-12. So ideally, you would like to arm the CAR-T cells. You can improve efficacy, but not increasing the toxicity. And this is what we would like to address then, if we can do that by arming our CAR-T cells with NAP. Because again, we are not focusing so much on helping the CAR-T cells. We rather would like to help the innate or the endogenous adaptive immunity in the patient to boost the response. So we have shown that this is actually the case in mouse models, and I'm not showing any data from it, but I'm showing the principles here. So this is then a tumor with heterogeneous antigen expression, illustrated by different colors of these tumor cells. So CAR-T cells armed with NAP, they would, just like conventional CAR-T cell, kill tumor cells expressing the CAR target. So we have killing here. But then we also have signal transduction leading to release of this. So it's an inducible promoter leading to the release of NAP in the tumor microenvironment. And that leads to recruitment of innate immune cells. We have monocytes, DCs, NK cells, and neutrophils. And some of those innate cells, like neutrophils and NK cells, they can take part in the killing of the tumor. And this killing is, of course, completely independent on whether the tumor cells express the CAR target or not, because this is a different killing mechanism. And maybe even more important is that we recruit dendritic cells and we activate them to phagocytose whatever is being released from these tumor cells. Of course, some 99% of this is going to be self-protein, and you're not going to be able to induce any immunity against that. But there will also be neoantigens released here and shared tumor antigens. So without having to sequence and knowing about those neoantigens, we hope that by having NAP secreted, we can tease that out from the tumor. And dendritic cells can take it up, and we know that NAP is inducing the migration of dendritic cells to a draining lymph node, where they can present these neoepitopes to naive T cells, and you can have activation of T cells with a T cell receptor that is matching the neoepitope. And those T cells can come back and start killing tumors in an HLA-restricted manner. So by adding NAP here, we have bystander immunity in form of innate immune cells and of T cells. And this one is also then referred to as epitope spread, because now we are targeting other epitopes than the CAR targets. So presumably the neoepitopes here. So it works in animal models, and we have now started also clinical trials running this. So the first trial is for lymphoma patients, and there we decided to develop CAR-T cells against CD20, and we did that so we should be able to treat also patients that are relapsing after conventional CD19 CAR-T cell therapy, and also for patients that are not eligible for those kind of therapies. So we have developed a production in the Prodigy and made the tech transfer to the GMP lab. And so when the patients come into the hospital, they go for apheresis, and we send then the white blood cells to the GMP lab, where the T cells are being activated, transduced with the retrovirus. And this retrovirus carries the CAR, but it also carries the cassette for the NAP expression. We do expansion, we formulate, and we cryopreserve. We are required to do cryopreserving at this point to make a quality control before they are being released. But we have 17 days from vein to vein. And these patients then, in the meantime, can have bridging therapy, and they get lymphodepletion before CAR-T cell infusion. So we are running a phase 1 trial, dose escalation. We have today treated the 9th patient last week, but we have elevation from 8 patients so far. And just to illustrate how this can work, I will take the very first patient that we had. You can see here that when he comes in for leukapheresis, we analyze the cells in the Luka product. You see staining for CD14 monocytes here. So we have the B cells over here, you have the T cells and the NK cells over here. So you see about 3% of the cells in the Luka product were B cells. We make CAR-T cells and give them back to him. And then we can see that all the normal B cells are gone after 11 days. And we can also see that if we stain the T cells, even 25% of the T cells in the circulation of this patient are the CAR-T cells now. And the dose here was 2 × 10⁵ CAR-T cells per kilogram. There was a pretty big guy, so maybe 100 kilograms, so he got 20 million CAR-T cells then. But after 11 days, we go from 20 million to 25% of the entire T cell pool. So a huge expansion. And this is enough to lead to complete remission. So this is the screening before surgery. This is physiological uptake in the heart, but you have the tumor over here and down here. And after 1 month, he was in complete metabolic remission. And he remains in complete remission. So he was treated back in November 2024. And in November 2025, then he was still in complete remission. So out of the 8 patients that we have evaluated, 6 went into complete metabolic remission after 1 month, and 4 of them are remaining in complete remission. And the longest remission we have done is now 1 year. The idea, of course, with this phase 1 clinical trial was to see if NAP is adding any toxicity to the treatment. And we haven't really seen that. We have no grade 3 or grade 4 CRS or ICANS. So it looks to be safe in this setting, which is important for us, meaning that we can continue with other cancers as well. This is just from our local newspaper when the patient, well, here he was in complete remission for 9 months, so they wanted to make a reportage. And these are the clinicians, Gunilla and Mats, treating him. So when it comes to CNS lymphoma, the phase 1 trial was with adult patients without any CNS involvement. So we are thinking of starting also a trial with CNS lymphomas. And we were also not allowed to include children in the dose escalation trial. So we are also thinking about that one and setting up hopefully a pivotal phase 2 clinical trial as well. But the real aim with arming CAR-T cells with NAP is to help in solid tumors to counteract antigen heterogeneity, the problem of targeting just one antigen that you can then induce this bystander immunity. And our plan is to go for glioblastoma. And the target antigen that we have chosen is IL-13 receptor alpha-2, which has been validated by others as being a good target, at least for glioblastoma. It is heterogeneous when it comes to expression, so it's going to be difficult to kill all the tumor cells by just this one target. When it comes to glioblastoma, most studies are targeting either IL-13 receptor alpha-2, which is considered a cancer testis antigen because it is re-expressed in many malignancies, including glioblastoma. Another good target is, of course, completely tumor-specific, the truncated EGF receptor. That would be a completely tumor-specific target. And both of them can be used, even tandem CARs can be used. And here you have the study by Marcella, when they're also arming their CAR-T cells with a bispecific antibodies secretion. So in all cases, therapy seems to be safe. We have responses, transient responses, but tumors come back, and this can also be seen in this case report from Christine Brown when they treated patients with IL-13 receptor alpha CAR-T cells. This is before treatment, and this is after treatment. So the CAR-T cells they have killed whatever tumor cells they can kill, but the cells that did not express IL-13 receptor alpha-2 were the clone that kind of grow back afterwards. So we think here that it's then even more important to have this bystander immunity, and that's why we would like to test this for glioblastoma. Others have shown that CAR-T cell infiltration into the CNS tumor is a problem, and CAR-T cell persistence is a problem. So that is typically being addressed by local injection and by doing repeated injections, typically into the ventricle. And then we hope to address the issues with immunosuppressive microenvironment, and especially the target antigen, the heterogeneity of the target antigen by arming our CAR-T cells with MAP. So we developed our own single chain against IL-13 alpha-2 using a national resource, SciLife Lab, that have phage display, and we could find a good antibody. We screened this antibody with the help of retrogenics to show that it is completely specific. There is no cross-reactivity with IL-13 receptor alpha-1 or any other human secreted or plasma membrane protein. So it seemed to be a good antibody that we're using for the CAR. We have started to run mouse models. We can see efficacy also in these mouse models when we inject locally. So there is efficacy. We see a transient activation of microglia in these studies, and that can, of course, relate to toxicity, but it can maybe also be beneficial if the microglia is attacking the tumor cells. So that remains to be seen. But we want to set up this clinical trial, and the way we envision to do it is to treat recurrent glioblastoma, ideally at their first relapse, and that we confirm that they are IL-13 receptor alpha-2 positive. We will make this screening from the primary resection. And when they come back for a relapse, we will do this leukapheresis, collect the CAR-T cells, and make and produce the CAR-T cells. And then when the patients go for removal of the resurgery, removing of the relapsing tumor, the surgeon will also implant an Ommaya reservoir with a catheter into the lateral ventricle. So we can then repeatedly treat those patients. So the standard would be coming in at relapse, making the CAR-T cells, surgery, implantation, and then treatment. There is, however, a great theoretical advantage of treating even before surgery, and that is because NAP wants to tease out the neoantigens that we have there. So we kind of need a tumor material to start with for NAP to be active on. So we would like to be able to treat also a few patients before their resurgery. And I'm bringing this slide up because I like to get comments from Tobias and Michael about this, whether this is feasible or not. So we don't want to implant an Ommaya reservoir before the resurgery, because that would mean 2 brain surgeries close in time. So we are then instead thinking of an intrathecal injection for a pre-treatment. And we do that because we will then, when the surgeon take out the tumor, we will then have the material, and we can look for mode of action to see, we can look for clonality of T cells if NAP has been able to attract an endogenous T cell. So we get this mode of action. And it is also, again, a theoretical advantage because we have tumor material left in this case for NAP to act on. So I'd like to get comments on this and discuss that maybe after the presentation. So this is where we are. We have treated, we have started to treat 8 patients in our first trial targeting lymphoma, and we are opening the glioblastoma trial, we believe, after summer. And these are some of the people that are working on this. How am I doing with time?
If you have more, otherwise you could conclude. But
I can conclude. I can conclude with it. Give me 2 minutes. I want to address new things, the side effects that we are also working on, and you know that CRS and ICANS are problematic but manageable, but they're also very costly. These are American figures, but it's probably relatively similar in Europe. So to address this, we have focused on the transmembrane domain of the CAR, because it has been relatively neglected, thinking that this is anchoring the single chain to the cell membrane and connecting to the signaling domains. So we have been working on the transmembrane domain. So Stefano Barbera, a postdoc in the lab, who will also go to Maria's lab after summer, has been working on different transmembrane domain and saw that dependent on the stretchability of the transmembrane domain, the CAR was able to enter lipid raft or not. And what he found, and that puts the CAR in different locations on the cell membrane, and it's mainly based on the fact that we know that CD3 and CD45 are being separated when you form an immunological synapse, and that CD45 can dephosphorylate the ITEM motifs on the T cell receptor and also on the CAR. So by having a transmembrane domain from CD45 on the CAR, you will remain not in the core of the immunological synapse, but on the outside. And a CAR with CD45 transmembrane domain could potentially dimerize with endogenous CD45, and you could have quenching of the signal at this place. And this is what we see when we make either Kymriah or Yescarta with a different transmembrane domain. We have a reduced signaling, a reduced activation. This leads to reduced cytokine production from the CAR-T cells, both in vitro and in vivo. And we are thinking that this could potentially lead, by going from a conventional transmembrane domain to one from CD45, we are quenching the signaling, quenching the phosphorylation, and we have less cytokines, less interferon gamma, less TNF, lower CD40 ligand expression, and meaning lower expression of macrophages, which are the ones that are responsible for the CRS syndrome, and also lower, maybe hopefully also lower expression of the ICANS. And by dampening the signaling of the CAR, we can potentially also have reduced CAR-T cell activation and exhaustion. So I would like to summarize by saying that by arming CAR-T cells with NAP, we believe that we can address antigen escape in hematological malignancies, and hopefully also antigen heterogeneity when it comes to solid tumor. And by changing the transmembrane domain, it remains to be seen, but in animal models, it looks like we can actually reduce toxicity and hopefully also improve the quality of the CAR-T cells. So with that, I stop and I take questions.
Yeah, thanks a lot. Great concepts. The discussion for the glioblastoma trial we do separately. I would suggest time for one burning question, one short answer. Something? Yeah, over there.
Thanks for the talk. I'm over here, sorry.
Yeah, there. See.
So I'm not an immunologist at all, but I would have imagined that we would generate antibodies against NAP. So like in terms of like long-term remission, would this work, or do we not generate antibodies against it? Or?
You will generate antibodies against NAP. Also individuals with H. pylori infection generate antibodies against NAP. Before getting into the clinical trial, we induced NAP antibodies in mouse models and treated with the CAR-NAP T cells. It doesn't seem to affect the efficacy that much. I am more concerned that NAP will be expressed in the T cells and that the CAR-T cells will be targeted and killed. But hopefully that will happen after we have been able to induce bystander immunity.
Great. We move on, if that's okay, and postpone the discussion for the coffee break.
We are not so late, but we welcome the next speaker, Professor Christian Buchholz from the Paul-Ehrlich Institute in Langen, Germany. And Christian is going to talk about current and future concept of in vivo generated CAR immune cells.
Yeah, thank you very much. And also thanks a lot to the organizers for inviting me. It's always a pleasure to come to Zurich, especially since I spent my first postdoc here decades ago, meanwhile. So I will give you an overview on the recent events in vivo CAR-T cell therapy. A lot happening there. Before we come to that, we are always following, of course, at Paul-Ehrlich Institute, where we are responsible for authorizing clinical trials and also market authorization for biological medicinal products, how the field evolves and its booming times for CAR-T cells. And not only CAR-T cells, but gene therapy in general. What you have to take from this is that we distinguish ex vivo and in vivo gene therapy, and important for my presentation is the vector types. So ex vivo gene therapy is basically lentiviral vectors, and in vivo gene therapy is AAVs. That's the two important vector types we currently have in gene therapy in products that are on the market. For CAR therapy, we have these listed products, so basically against multiple myeloma and against B-cell lymphoma. And the number of clinical trials is continuously rising. So that's the numbers worldwide. And although it's only going until 24, the numbers are continuously rising also by the end of last year. Now, part of the clinical trials, of course, there are many open questions, and we heard here in the meeting a lot of them, but part of the clinical trials is addressing this question: how can you reduce the immense cost and efforts to generate for each patient a individualized GMP manufactured medicinal product? And there are different strategies ongoing. One is the allogeneic CAR-T cells. That's a strategy which is already in clinical trials for quite a while. And the concept here is that you have a donor T cell that is then engineered not only by CAR delivery, but also engineered, for example, to reduce the rejection by knocking out HLA class 1 or also checkpoint inhibitors. And this is then can then be given into multiple patients. Not all patients, you still have some HLA restrictions, of course. But so far, we don't have any product here that has successfully gone through phase 3 and have to see how that further develops. Another recent strategy is explained here. That's short-term CAR-T cells. That's still an autologous medicinal product, but you have substantially reduced the time to expand the T cells. So basically, you add the vector, and on the next day or on the second next day, already you apply this to patients. So reducing part of the problem, but of course not all of it. And what's going probably to the most far end of tackling this problem is the strategy of in vivo CAR-T cell therapy, which suggests that you generate the CAR-T cells directly in the patient, in the organism. And condition for that, of course, is that you have vectors that are highly specific for T cells and will not deliver the CAR to non-target cells, especially as long as we don't know what consequences that might have. And as I mentioned, this is currently coming up tremendously because last year we had the first data published from ongoing clinical trials. And the other important issue to mention is that there's really a lot of biotech companies. This is only a fraction, especially since all those companies that are active in China are not listed here. So biotech is really booming on in vivo CAR-T cell therapy, and they are using different strategies, basically different vector types to accomplish the in vivo delivery. So one company is Interius, and they were the first to announce a clinical trial starting in October 24, and that was a CD7 targeted lentiviral vector encoding CD20 CAR against B-cell lymphoma. And the interesting part, of course, it's expected cost of below $10,000 US, what they suggested, which compared to probably something like 300 to 400,000 euros per patient, which we have for ex vivo CAR-T cell therapy. And this trial was also authorized in Germany at the beginning of last year. Now, it's of course for us quite pleasure to see that basically all this goes back to this landmark publication from my lab, where we showed in 2018, and the experiments for that were sort of done maybe around 10 years ago from now, where we have shown that you can generate lentiviral vectors that are targeted against subtypes of T cells, CD8, CD4, also CD3 lentiviral vectors. And when you deliver a CAR with these vectors into mouse models, then you generate CAR-T cells directly in vivo, and these CAR-T cells will eliminate the tumor cells. You can see this here, where we have the luciferase labeled CD19 positive tumor cells, which mainly go to the bone marrow. And when you then have a look in the bone marrow for these tumor cells in the control mice, we have them here in this population, and it's basically eliminated upon a single injection of the CD8 targeted lentiviral vector. And what is, in my opinion, really important is the selectivity of these vectors for their target cells. And you see this here on the protein level. So this is the CD8 positive T cells, this is the CD4 positive T cells, this is the CAR expression, and there is basically all activity in the CD8 on-target population, whereas a conventional lentiviral vector cannot discriminate. And we have recently also followed this selectivity by single-cell transcriptomics, And again, you see here in this UMA plots the difference. This down here is the CD4 T cell population, which is spared out here. And when you then go through cell by cell, you can really conclude that there is more than 99% on-target delivery with these vectors. This is just in the presentation for your information overview of those companies and products that are currently in clinical trial, either with lentiviral vectors or with lipid nanoparticles. And I mentioned that there were first publications now out on clinical trial data. And this year is the trial in multiple myeloma patients with lentiviral vectors delivering the BCMA CAR. And you see here in this figure the viral particles detectable in the patient. So peak time by 6 hours, that's probably something you'd expect. And then within the next hours, the vector particles are cleared from the system. And this is the kinetics of the CAR-T cells. And you see that this peaks around day 10 to day 12, actually pretty much in agreement with what we have seen in the mouse models where we injected the CD8-targeted lentiviral vectors. 4 patients treated in this trial, 2 complete response, 2 partial response, and very much to stress is this is without lymphodepletion. Of course, lymphodepletion would eliminate the T cells, which are the target for the lentiviral vector. No conditioning, but yet you have CAR-T cells levels which are pretty much in the range of what you have when you do conventional ex vivo CAR delivery. And apart from this publication in Lancet, there are also meeting reports. Here, one at ASH from another company also delivering BCMA CARs in multiple myeloma, and very much the same outcome, I think, also for 3 or 4 patients in this case. And there's also meeting reports on B-cell lymphoma and CD19 targeted CARs, where it also looks that there is benefit for patients. But of course, all this is very short observation periods of a couple of months. That was all lentiviral vectors. There's also one publication came out on lipid nanoparticles delivering the CD19 CAR, not in cancer, but in autoimmune disease, that is lupus. And what I'd like to draw your attention to here is this extreme kinetics. This is the percent of CAR-T cells in the periphery. And you see that we come here in this patient to values of over 50% within 6 hours after injecting the lipid nanoparticles. And simultaneously, this is the CD19 B cells going down again also with this enormous kinetics by 6 hours. And this is then, of course, requires repeated administrations, because with the RNA you only have transient CAR expression in contrast to the lentiviral vectors. So having gone through these clinical data, we now switch to the vectors and vector engineering. And as mentioned, we have the lentis and the lipid nanoparticles currently used for in vivo CAR delivery, which is sort of contrary to what we see in the products on the market, which is basically AAV vectors for in vivo gene therapy. So they are, of course, so far underdeveloped for CAR delivery, and that is, although they are the most used vector for in vivo delivery, they have a known convincing safety profile. There's no risk of insertional oncogenesis, also no risk of incorporation of the CAR into the particles. They are known from intracerebral injections to have very nice diffusion away from the needle track. You can have targeted genomic integration when you combine with, for example, CRISPR-Cas immunonucleases. And we've also shown that you can make bispecific AAVs for two target receptors, which expands the level of possibilities. So how is the engineering process done to achieve this high level of on-target activity? You need two things. You need to destroy the natural receptor recognition by point mutating residues that attach to the natural receptor. And you then have to add a high affinity binder for the target receptor of choice on T cells, for example, CD8 or CD3. And that's something we initially did for lentiviral vectors on glycoproteins in the lentiviral envelope, and later on also in AAV vectors where we have a non-envelope particle, but where you can manipulate the capsid and also insert binders into the capsid. And most recently, I will explain you also that we have also initiated a strategy to target lipid nanoparticles via ApoE DARPins. This is an overview on the three different vector types we are using in my lab. Here are the targets on immune cells, and we are also interested in targeting tumor cells, which can be of interest when you want to use targeted vectors, for example, to manipulate the tumor microenvironment. So a few slides on AAVs targeted. The first publication, so this is the capsid protein in AAV. And at its N-terminus, we initially fused the DARPin that in this case does not bind T-cell marker, but glutamate receptor, which is expressed on interneurons. And you see that this worked, but on a hippocampal slice culture, we had a relatively moderate activity. And more recently, we have switched the insertion site into a loop region over here, where we now insert the DARPin, and by that we have achieved a extremely increased gene transfer activity. And now we can also see in high resolution microscopy the DARPin on the surface of these AAV particles. And when you use this for T-cell targeting, and here is an ex vivo experiment where you mix the particles into donor blood, and then 6 hours later isolate PBMCs and analyze them for gene delivery. This is the parental AAV not targeted against T cells. And you see that by CD8 targeting, we achieve a much better gene transfer and also a high selectivity for the CD8 positive T cells. And this also looks promising in humanized mice, where we also have a nice selectivity of gene delivery into the CD8 positive cells. However, that's at the moment the drawback here. We need to activate the T cells in this mouse model by injecting IL-2, T3, but it can't be working on further engineering steps to get rid of that. With the LNPs, I mentioned that we have come up with a strategy based on apolipoproteins. So here on the left, you have an overview on targeting strategies for lipid nanoparticles. And this morning, we also heard about another one. Usually, when you do the active targeting with antibodies, they are chemically coupled to lipids. And what we have here is a rather biological approach where we fuse the binder, so the CD8 specific DARPin, to apolipoprotein E, and this is then expressed in E. coli and added to the lipid nanoparticles. And the thing you have to know here is that there are different types of APO, of course, and they all bind the LDL receptor as the target. And LDL receptor, for example, is highly expressed in liver, which in part explains their liver tropism. And the affinities for LDL receptor are especially high in ApoE3, lower in ApoE2. And the issue here is that by mutating residues that bind the LDL receptor, we can now use this detargeting strategy that we use in the viral vectors, also here in the lipid nanoparticles. And how that looks when you go to primary human T cells, so add the lipid nanoparticles. When you don't add any ApoE, there is basically no activity because the particles do not have the corona. No apoproteins cannot bind LDL receptor. When you add ApoE2 expressed in E. coli or ApoE3, you see that you now have binding of the lipid nanoparticles to the CD4 as well as the CD8 T cells, no discrimination, but it nicely shows how these particles depend on corona formation. And when you then have ApoE2 CD8 targeted, you see that we have the CD8 population with a higher signal than the CD4 population, but there is still activity on CD4. And when you then mutate the LDL receptor binding, you get rid of this. And it also looks pretty similar when you now use whole blood, where these particles are mixed in. Again, we have now some activity when we have the ApoE2 added, but you see here this strong CD8 binding when we have the ApoE2 CD8 fusion protein formulated onto these particles. And the last slide is now the combination of lipid nanoparticles and AAV vectors, because the AAV vectors will not integrate the CAR into the genome, but when you deliver with the lipid nanoparticles, the Cas9 and the guide RNAs, you can then have the CAR integrated into the T cells. And you see that due to the CD8 targeting of the AAVs, we have the CAR expression selectively in the CD8 positive cells, and this is combined with down modulation of the TCR because the CAR is delivered into the track locus. So that could be an approach to use in future then also AAV particles for in vivo CAR delivery. So by that, I come to the take-home message. So in vivo CAR delivery has entered clinical trial, which suggests successful CAR T-cell generation in patients. And T-cell targeted vectors are key for this. DARPins are ideal for receptor targeting. They can be selected for any receptor of choice and even discriminate between closely related cell surface molecules, something I haven't time to show you, but we can do counter selections in our DARPin selection strategies with receptors you don't want to bind against and thereby be very specific for target proteins. Recent developments of AAVs expand the repertoire for in vivo delivery, and the fusion of DARPins to ApoE enables a biological approach. And with that, I thank the funding and collaborators in my group and you for your attention.
Thank you very much for the presentation. The talk is open to questions. I don't see. So you discussed, I have a question about activation because you haven't mentioned a lot, but in your preclinical model in the mouse, you need to activate T cells, right? So they incorporate the transgene. What are currently the main advancements to deliver in vivo and simultaneously activate T cells?
So this requirement for activation was when we use AAV vectors for gene delivery. You don't have to activate when you use lentiviral vectors. So that worked also in humanized mouse models where you have reconstituted with CD34 stem cells, and where it's known that the T cells in this mouse model are not activated due to the xenoreaction, for example, but with the lentiviral vectors, you can also get successful delivery in that scenario.
Any additional question?
Thanks a lot for the nice talk, Christian. Just one question. So definitely we're seeing that the floor is heating up a lot for in vivo CAR-T. I think mainly is a challenge of delivery. Do you see any winner between lentiviral or non-viral mRNA LNP modalities?
Yeah, it's too early to conclude, but from the expectation, of course, is that for cancer treatment, I would say that you need a persistent CAR expression, and therefore I think that can only currently be accomplished with a lentiviral vector, whereas for the autoimmune example that we saw, where you sort of need to restart the immune system and may already have benefit for the patient when you have a one-time elimination of the B-cell repertoire, that could be the best strategy for the lipid nanoparticles.
Existence according to the modality, that's a bit what you would suggest.
At least from the current view, but I think the field is evolving rapidly, and but there are, of course, also scientists saying that with the lipid nanoparticles you can do repeated applications, therefore they may also be useful for cancer treatment. There, of course, the question is how is the safety over time? I mean, that's in general, of course, the question for all the in vivo approaches, how is the safety over time? But in addition, the question also is, will you at one point develop a immune response against the vector particles? And probably also the lipid nanoparticles are not completely inert for that.
Thank you. Just a short question.
Yes. Has anybody tried, even in vitro setting, or better in vivo, to do a re-challenge? So and see if are the same cells being engineered, or so a re-challenge with the virus. So delivering.
I mean, that's what I showed in the clinical trial with the lipid nanoparticles and the autoimmune patients. There you had, I think, 5 or 6 repeated applications of lipid nanoparticles in over every 3 or 4 days. Yeah?
But we don't know if it's the same cells. So
well, if what I showed is that you hit more than 50% of the T cells in the periphery with one shot. So I'm sure that will be repeated. It's
And so to give the platform now, we have Professor Sonja Turges, who is professor at the University of Heidelberg and Mannheim. And luckily, she's also still a little bit at the University of Zurich. So we'll be at our institute. And she'll be talking about the lymphocyte networks in colorectal liver metastasis.
Yes, thank you very much, Burkhard, and also thank you very much to the organizers for the invitation. It's a great meeting, and I'm happy that we can present some of our research here, which has to do with lymphocyte networks orchestrating chemotherapy response in colorectal liver metastases. And colorectal cancer is a leading cause of cancer-related mortality worldwide. And this is largely due to its frequent progression to metastasis, which occurs in about 50% of colorectal cancer patients during the course of the disease. And in these advanced colorectal cancer patients, the liver is the main metastatic site. And this is usually associated with poor prognosis, with a 5 years overall survival usually of below 40%. And this is only if these tumors in the liver can be resected. So liver resection offers the best chance of long-term survival and cure in these patients. And therefore, these patients are usually treated with neoadjuvant therapy, which mostly consists of chemotherapy, which is regimens of FOLFOX and FOLFIRI, and sometimes also targeted therapy. And this is done to downstage the tumor in order that these patients can undergo surgery. However, only 15–30% of these patients respond to neoadjuvant therapy, and therefore there's a strong clinical need to understand the mechanisms behind this. And this is partly due to the challenges in inducing anti-tumor responses in the liver tumor microenvironment. So about 95% of colorectal liver metastases are microsatellite stable, and this means that they have an intact DNA repair system and therefore a low neoantigen load. On top of this, the liver is a very tolerogenic organ because it is naturally programmed to inhibit immune responses, and this is actively hijacked by a very immunosuppressive tumor microenvironment, which impairs the function of dendritic cells and anti-tumor T-cell activity. And because of this, colorectal liver metastasis has been historically shown a very poor response to immune checkpoint blockade, and to date, there's no immune-based therapies approved for this patient population. I'm talking about the microsatellite stable patients, and chemotherapy is the standard care. However, there's still some hope, because despite this orchestrated immunosuppression, T-cell infiltration is a strong predictor of outcome in colorectal liver metastasis. And this is exemplified here by the immunoscore, which is the quantification of these T-cells in the tumor center and also in the invasive margin, with high densities of CD8 T-cells correlating with better survival. However, the CD8 T-cell metrics alone fails to capture the full complexity of immune regulation, and this is because these tumor-infiltrating lymphocytes are not acting alone, but they operate in these distinct microenvironmental regions within the liver, where the interactions with the surrounding cells are going to shape their function and fate. On top of this, chemotherapy can reshape the T-cell activity in the tumor microenvironment. Oxaliplatin, for example, which is a main component of this regimen FOLFOX, can induce immunogenic cell death through the release of danger signals, and this can lead to dendritic cell activation and enhanced T-cell priming. However, irinotecan, which is another a component of this other regimen here, FOLFIRI, has been shown to inhibit some of these immunostimulatory effects. So ultimately, the success of chemotherapy depends on whether these changes will translate into a functionally competent anti-tumor immune landscape. And this raises a fundamental question: which T-cell programs and in which spatial niche orchestrate the response to neoadjuvant chemotherapy in colorectal liver metastases? And to answer to this, we initiated a study together with Bettina Sobotka from the Department of Pathology at the University Hospital in Zurich. And this study was led by Maud Mayou, a very talented senior scientist in our laboratory. And to map T-cell states linked to neoadjuvant response, what we did was to collect 30 liver resections from colorectal liver metastasis patients who underwent neoadjuvant chemotherapy before surgery. And we separated these samples into these three regions: the tumor center, the invasive margin, and a more distal part, which corresponds to the perilegional normal tissue. And we took these tissues to perform high-dimensional flow cytometry to identify T-cell subsets and their activation status, which we can later correlate with the pathological response to chemotherapy, stratifying the patients into responders versus the non-responders. And this is the resulting UMAP of the different lymphocyte subsets that we obtained. And when we quantified these across the different regions, you can see a striking exclusion of cytotoxic cells, NK cells, gamma delta T cells, and also mite cells in the tumor center, which is rather populated by some CD8 T cells, but mostly CD4 T cells, which constitute about 60% of the total lymphocytes in this area. We then zoomed into the T cells to resolve T cell subsets and their activation status. And for CD4 T cells, we found several populations of memory cells, some of them expressing the activation residency marker CD69, while the others rather feature the stem cell marker TCF1. And for CD8 T cells, we could also identify several populations of memory cells, here positive for CD45RO, but also different shades of effector cells, which express distinct levels of CD69, granzyme B, KLRG1, PD1, and many others. And instead of looking at all these populations separately, we asked the following question: which cell populations show correlated abundance across regions? And to answer to this, we correlated the lymphocyte. We correlated the lymphocyte percentages, and we performed this hierarchical clustering to retrieve these modules of cells that vary together. We then correlated these modules with across the different tumor regions and per patient, and we found this module here, which is basically composed of innate and effector T cells, which was very prevalent in the perilegional normal tissue, but was absent in the tumor center. And then there was also this big module here, composed of mostly memory T cells, but also some exhausted cells and Tregs. And this one was the dominating module in the tumor center. And the next step was to correlate these T cell populations with a response to neoadjuvant therapy. And for this, we used this tumor regression grading, which is the histopathological classification system based on the proportion of tumor cells and fibrosis in these livers. And a tumor regression grade low means response to chemotherapy. And as you can see here, this is associated with better outcomes. And a tumor regression grade high means lack of response to chemotherapy, still a big tumor burden, and this is associated with worse clinical outcomes. Got blocked. Yeah. That's right. And again, we performed the same module analysis than before, but now focusing on the tumor center to try to find T cells that co-vary together in this region. And we find these 5 distinct modules. And in the non-responders, you can see that the more prevalent module is this module metastatic 3, which is driven by a population of CD4 memory cells, which we call memory 1, and also regulatory T cells. And this is very different in the responders, which are basically dominated by this module memory MET4, which is basically composed of CD4 memory 2 cells, and also 2 populations of memory cells, the CD8 memory and the CD8 effector memory cells. And what are these cells? What do we know about them? And this is a simplified picture of different hints that we obtained by flow cytometry. And if we focus on the populations of CD4 memory cells, what we can see is that the most distinguishing marker is that in this population of CD4 memory 2, which is associated with response, we have a high expression of this activation marker CD69, whereas in the memory 1, which is associated with a lack of response, we have just a bit more expression of the stem cell marker TCF1. And also for these 2 different populations of memory cells, we found some distinguishing markers. And even though this gives us some insights, we still wanted to get to know more about these cells. And to do that, we used the Rhapsody platform, which combines antibody sequencing and single-cell RNA sequencing. And this allows us to link cell surface markers with the transcriptome for each of the cells. And doing this, we could map back all our CD4 T cells that we had previously identified by flow cytometry, including the memory 1 and the memory 2 cells. And when we looked closer at their transcriptome, we found that the memory 2 cells associated with response express markers of activation and differentiation, but mostly markers related to Th1 polarization, including CD4 T ligand, TNF, and some interferon gamma-related genes. In contrast, the memory 1, which are associated with a lack of response, show a more circulatory and less activated profile with a high expression of genes related to the TGF-beta pathways and the inhibition on MF-kappa beta signaling. And consistent with these profiles, when we took these cells ex vivo and we stimulated them with anti-CD3 and recombinant CD40, we could see that this CD4 memory 2 displayed a a Th1 biased, which you can see here by the upregulation of Tibet, CD40 ligand, and some other activation markers. And these cells also produce more cytokines, including interferon gamma, TNF, and also IL-2, in comparison to the memory 1 subset, which is associated with the lack of response. And using the same platform, we could also identify the CD8 subsets associated with neoadjuvant response. And here we have first these memory cells, which express many genes related to stem-like and central memory cells. And this is very reminiscent of this stem cell memory population described by Gattinoni and Lugli as a reservoir for long-term anti-tumor immunity. And we also have this population here of effector memory cells, which shows a progenitor-exhausted-like profile with a high expression of this effector molecule here, this granzyme K. And this has been shown as a transitory state which retains progenitor potential, but can differentiate towards effector cells when receiving the right signals. And when we stimulated these cells ex vivo with anti-CD3 and anti-CD28, you can really see that the effector memory cells got very activated. However, these cells produce less cytokines in comparison to the stem-like memory CD8 T cells. So now we have characterized much better these T cell programs that are associated with response to neoadjuvant chemotherapy, with these Th1 CD4 memory cells providing some help, the stem-like CD8 memory cells acting as a responsive reservoir, and these memory CD8 T cells that are positive for granzyme K retaining effector potential. And the question is that to get to know a little bit more about these cells, we need to understand where they are and how these cells are organized within the tumor microenvironment. And to do that, we performed multiplex immunofluorescence, and we did that with tissue microarrays from these 30 colorectal liver metastasis patients using punches from different areas. And we designed a panel that allows us to detect tumor cells, but also different types of antigen-presenting cells, as well as activation markers for these T cells associated with the therapy response. And what we found is increased immune infiltrates, as you can see here in the invasive margin, and this is mostly dominated by CD4 T cells and myeloid cells. And in the tumor center, even though the infiltration is sparser, we found increased densities in the responders of both CD4 and CD8 T cells. We then performed cell-cell proximity assays, and what we found was that in the tumor center of the responders, the CD8 T cells were localized much closer to dendritic cells and also to CD8 T cells, mostly the ones expressing this stem cell marker TCF1, and also some of them the effector molecule granzyme K. And these cells were not just localized randomly, they were forming some kind of organized structures, as you can see here, and we called these structures therapy-responsive immune niche, or TRINs. And within the TRINs, we could quantify the triads of CD4 T cells, progenitor CD8 T cells, and antigen-presenting cells, as you can see very nicely here. And we found that these three-way interactions were much more abundant in responders, both in the invasive margin and in the tumor center. And the difference between these two regions lies on the composition of the antigen-presenting cells. Because in the tumor center of responders, the triads are mostly composed of dendritic cells and MHC class II positive macrophages, whereas in the invasive margin, these triads can also contain B cells. So what we think is that CD4 T cells play a key role in shaping productive anti-tumor immunity in colorectal liver metastasis patients responding to chemotherapy. And this is not about the amount of CD4 T cells that we have, it's about their activation status, and it's also about their spatial localization within this therapy-responsive immune niche. And in responders, these Th1-like memory cells colocalize with specialized antigen-presenting cells and also with some progenitor memory CD8 T cells, and we think that this organization allows them to license these antigen-presenting cells, probably through this CD40 ligand CD40 axis, And this induces the reactivation of CD8 T cells and enhances response to chemotherapy. However, the truth is that we still know very little about these streams. So what we have done now is to initiate a more integrative study where we have cohorts not only from colorectal liver metastases, but also from the matching primary tumors. And this we do with Bettina Sobotka and also with our collaborators from the Charité. And what we do here is to study the cellular molecular composition as well as the gene expression programs of these streams. And we try to correlate this with the tumor intrinsic mutations in the tumor and the clonality of the T cells in situ. And with all these insights, we would like to provide prognostic associations with patient survival. But the most important thing would be to predict the response to this neoadjuvant therapy from the primary tumor, and also to identify some actionable tumor immune targets for future therapeutic strategies. And last but not least, we are trying to harness this CD4 T cell antigen presenting cell axis to enhance the chemotherapy response in this tumor. And to do that, we started working with patient-derived tumor fragments that Daniela is going to introduce later, and also with mouse models of liver metastasis based on organoids. And the first thing we do is to test the effects of different chemotherapy regimens to see which ones promote a better response for the CD4 APC CD8 axis. We also try to study the determinants of the formation of these memory T cells, and based on the results that we have from the patients, we try to find rational combinations of immunotherapy plus chemotherapy based on the in situ licensing of antigen-presenting cells, vaccination, and trying to reinvigorate the progenitor memory pools. And with this, I reach the end. I would like to thank the drivers of this work, which is basically Maud Mayu and Bettina Sobotka, all the students and collaborators involved, the funding agencies, and all of you for your attention.
Thank you very much, Sonia. We have some questions right away, Michael.
Yes.
Yeah, Sonia, very nice talk. So one thing, did you also have biopsies or the surgery piece after? Or only
so this is fresh samples, and but we process them exactly. So we do slow freezing with the samples, and then we do the analysis afterwards with the samples all together.
Oh, okay.
Yeah.
And
I mean, for the first part of the talk.
And the second question is, so I've seen Tregs multiple times in the non-responders. So did you look at those? Because there's evidence now in early phase clinical trials that if you have Treg depletion in the MSS patients who don't respond to immunotherapy, you might resensitize them.
Yes, I didn't talk about them so much today, but they definitely play a role, and we see them consistently, being let's say linked to the lack of response, right? Especially these effector Tregs, and we also know from another study now from the Kisik lab that if you remove them, some of these TF1 positive CD4 progenitor cells can actually differentiate towards a more Th1 profile. So I think it's going to be very important to remove them in this particular context.
Thank you very much for the talk. I'm curious to know if you have any information about the physical properties of the tumors in the responders versus non-responders, and if that could potentially influence the accessibility of the immune cells to form these functional niches within the tumor.
Meaning in the vessels or
density of the matrix?
Yeah, I don't know. Actually, that's a good question. So it's something that we are addressing a little bit more now when looking a little bit more broadly at this niche, right? Because right now we were focusing, of course, mostly on the immune part. But with this new integrative study, we are actually trying to look at the stroma, focusing also on the vessels and trying to see what are the effects of chemotherapy there, and it's also bringing this to the mouse models, because I think it's going to be very important also to determine the accessibility of these cells to the tumor.
All right, Daniela. Oh, sorry.
One very short question. Did you also have access to tissue from the tumor draining lymph nodes?
Oh, it's a very good question. Unfortunately, well, we didn't have it for these patients, but what we are doing now is a retrospective study with a primary tumor and then the local regional lymph nodes from this primary tumor, which we also analyze, and then we try to link it also to the liver mets, and also, of course, we are analyzing the lymph nodes in the mouse models.
Thanks a lot, Sonia. That was excellent. Thank you very much.
And over to Violeta Goveleva from Zurich, and she's talking about MS4A7 positive monocyte-derived macrophages in tumor immunity.
Thank you so much for introduction, and thank you for the opportunity to present the symposium. So this work was performed by the entire Becher team, and I'm presenting it on behalf of the group. Tumor-associated macrophages are the most abundant immune cells in many solid tumors and represent a major barrier to effective cancer immunotherapy. Clinical strategies targeting these macrophages have shown limited success because it still remains difficult to selectively eliminate pro-tumor immune suppressive macrophages while preserving macrophages with anti-tumor activity. To address this challenge, we performed single-cell RNA sequencing to profile myeloid cells in the mouse model of GL261 glioma during IL-12-mediated immune reprogramming. This analysis identified MS4A7 as a marker of a discrete population of monocyte-derived macrophages with an immunosuppressive signature characterized by co-expression with TREM2 and ApoE. And to study these cells in vivo, we generated a mouse model, MS4A7 Cree mice, for genetic fate mapping and conditional manipulation of MS4A7 positive cells. Across different tumor models, like subcutaneous MC38 or B16 or GL261 glioma, MS4A7 Cree-driven reporter expression was restricted to macrophages. And importantly, deletion of CSF1 receptor, a key survival receptor for these macrophages within the MS4A7 population, led to reduced tumor growth. Altogether, we identified MS4A7 as a potential marker of immunosuppressive macrophages and generated a genetic tool to study these cells in vivo. Thank you so much for your attention.
Thank you very much for this talk and the compact presentation, very clear. Any questions? The last question of the day. Yes, please.
Great, pleasure talk. I have a question. There is a drug available in the clinic, axatilimab, that targets CSF1 receptor. It's used in chronic GVHD. Any plans, or have you tested to target this population with an antibody or with a small molecule in order to be able to translate it into the clinic?
Thank you so much for the question. It's a great question. Yeah, we have an idea to also target these cells with the CSF1 receptor antibody. This is a potential avenue for research for us. Yeah.
Thank you.
Thank you very much once again. So last talk of the day. And after listening to all these great speakers, I will hand over to Markus Manz for closing the conference.
Yeah, thank you. So my wrap up is simple. I'd like to thank you all for being here. I think we have seen really, again, mind-blowing science. It's great how basic understanding shows us always the translation to the benefit of the patient. And I think the steps that are now taken forward are really immense, and it's a great pleasure to see that. So thanks to all the speakers for coming, for sharing their data. thanks for you to listening. Thanks to the people that engaged in lively discussions. That's always fun, and we found time to do that. So also thanks for sticking to the speaking times. Thanks for the organization again, and for having all the slides and so on, but one prepared. And thanks for the support without which we wouldn't be able to do these kind of meetings. So again, thank you all. Have a great evening, great weekend, and safe travels home. Thank you.